Good morning, everyone. Welcome to the University of Miami's Rosenstiel School of Marine and Atmospheric Science Diversity in Science event. I am one of your hosts, Ana Sangronis.
And I am your co-host, Aaliyah True, and I am a first-year PhD student at the Rosenstiel School in the Cnidarian Immunity Lab. And I'm super excited to have everyone here today.
All right. We are going to start out with a warm welcome from our Assistant Dean, Dr. Joshua Coco.
All right, welcome everybody. Super excited to have y'all here. I'm actually not even in Miami today. I'm actually over in Orlando at the Florida Undergraduate Research Conference. So hopefully some of y'all get the opportunity either in the next couple of years to get some opportunities to do some research in the field and have that option to go and present at a conference like the Florida Undergraduate Research Conference or a national conference. So y'all should definitely check into some of those, but we are super excited to have y'all be a part of our second annual Diversity in Science event today. This is something that we started last year and was super excited about. Had a great turnout. We're excited to see a number of y'all here today. Our Dean Ronnie Avasar, couldn't make it today. He's usually the one who gives the speech, but I know he sends his regards for that. I did want to do a quick kind of presentation, just so y'all can get an idea a little bit more of our school. So right here, we've got just a little bit of a background of the different types of students that we have over at the campus. So as you can see, we've got tons of international students from all over the world. And I know even today, we're going to have a handful of students who are from a country outside the United States. So we welcome you all as well to be here today. But in general, I love this 4, 3, 2, 1 kind of number setup we have here. So about 400 undergraduate students in our majors, such as marine biology and ecology, marine science, double major with biology, computer science, meteorology, etc. We've got our own separate major for meteorology, for geology, for oceanography, for marine affairs. So lots and lots of different options that are out there for students to partake in, going from high school into college. And then we've also got our graduate programs, which we've got with our PhD programs, with our Master of Science and our Master of Professional Science program. So, you're going to get a great opportunity to come to our campus and kind of get that hands-on opportunities that a lot of people are looking for before you go on to your jobs. And then we've got that 200 and 100 with the 200 staff and 100 faculty. So you get really good individualized attention and advising whenever you're part of our programs. So I always love this shot. This is our campus, right? This is the Rosenstiel School at its finest. You can kind of see us on the left side over here. You can see our research vessel, the Walton Smith. You can see our scientific dive pool where you get to go and learn some amazing scientific diving opportunities that you're going to need if you end up working for somebody like NOAA or the National Park Service. And as I speak of NOAA, we've got NOAA literally in these two buildings over here. So to be able to have the opportunity to do some work with NOAA, we even have a cooperative institute for marine and atmospheric studies. So a lot of our graduates end up actually being able to work with University of Miami and with NOAA through that program. And all the way in the bottom right corner over there, you can kind of see our hatchery for our aquaculture facility. So if y'all are interested in growing fish and being sustainable, we've got those opportunities as well. And then of course, the beautiful background with Miami, right? It doesn't get much better than that. If you get the opportunity to come and tour with us, you'll get to drive over the Rickenbacker Bridge over there, and it's just going to be one of the most amazing days. So kind of going on, I kind of want to talk about some of the outreach opportunities that we do as it relates back to diversity. We've got our Ocean Kids and Explorers, which we got a grant this year that we're going to be working with NOAA's Marine Sanctuaries, where we're going to bring some students in onto campus from underserved Miami communities. We've got our Impact program where we host 6-week annual summer camp with low-income first-generation college-bound students. We've got the USOAR program where we've got our undergraduate organizations who are doing some work in the K-12 classrooms related to science of the ocean, atmosphere, and rocks, which I absolutely love that USOAR name. You've got the Females in Natural Sciences, FINS, doing a lot of great work. We're doing around 1,000 high school girls who are part of their hands-on experience in marine science and getting opportunities to to take part in that research with sharks. We've got our Students for Students program where our graduate students actually get to go out and do some work in the K through 12 area, also with the general public. They're doing work related to, with hurricanes and tornadoes. And so if any of you teachers that are out there are interested in having some of those students come from either into the classroom or do it virtually, reach out to us and we'll definitely give you some opportunities to get some of those students in there and have those science conversations with your students. We host every single year our Women in Science events. We hosted that back in the fall. We'll continue to host that in the future. We're hoping to do both a virtual and in person this upcoming year. So if you're interested in that, you're in the local area, we'd love to have you on campus. And if you're anywhere out around the world, we'd love to have you sign up and attend that. We've got our National Ocean Science Bowl group, which we work with FAU every single year to host both over on FAU's campus and over on our campus. It's an amazing opportunity for Eastern Florida to kind of take part in that high school piece. And of course, we've got our shark research and field experience where we're working with middle and high schools classes to kind of join them and take them out on that trip, because we know that hands-on experience in the field is super, super important. And the other big thing that we're doing right now for those who are looking to go into graduate school, we've got our Rosenstiel Opportunity Award. So, we're working with our Diversity, Equity, and Inclusion Committee to give underrepresented Master of Professional Science students the opportunity to get up to 100% in tuition waivers. So some of these students are going to have opportunities to come to the school and not have to pay a dime for their tuition. We offer 4 of those every single year, and we're super, super excited that we'll be offering that coming up soon. If you are interested, those applications are coming up soon. They're due March 1st. So if you're interested, definitely reach out to us and let us know. Those have started since fall 2020 and they're not going to end anytime soon. So we're excited to offer those every single year. But yeah, that's everything that I've got here. I'm super excited to have you as part of our group today. And Anna, take it away from here.
Great. Thanks, Dr. Coco. That was a really fantastic overview of all that Rasmus is and all that it has to offer. And forgive me in the beginning, I was so excited to get started. I forgot to give a little context as to who I am. And you guys got to meet Aaliyah, so I wanted to introduce myself a little more properly. I am an alum of the Rosenstiel School of the MPS program in 2014. And I currently serve as the Florida Sea Grant Extension Agent, which is part of the University of Florida IFAS Extension Service here in Miami-Dade County. And if you all are still present, at the end of the day, you'll get to learn a little bit more about what that is. And one of the things I really like about this event is that it's a great opportunity not just to learn about people's different cultural and ethnic backgrounds, but also their different parts and walks of life and how they got to be in this field. So the presentations that we have lined up today are going to be a terrific learning opportunity and chance for engagement and ask questions of these researchers. So starting the day off, we have Dr. Amanda Ehlert, Dr. Martin Grossell, Dr. Rachel Hoyer, Dr. Eric Volkerts, and their student Jasmine Garza, who are going to be presenting to us about the physiology and biogeochemistry of fish carbonates. So go for it, everybody. Share your screen. Take it away.
Thank you, Anna. Here we go.
I'm not sure if we're supposed to be hearing something, guys.
Oh yeah, you're not hearing that?
No, you might just need to reshare and share your sound.
Okay, let me also take out my headphones. Maybe that will help. Letting me know.
Okay.
Thank you all so much for joining us
How's
today.
that?
We're going to be talking
Great.
about physiology and biogeochemistry of fish carbonates, or ichthyocarbonates, as we'll call them throughout the presentation today. So to kick off our talk, I'd like you to imagine a white sand beach. Here, I'm showing you an example of the beach right next to the Key Biscayne Lighthouse. And on that beach, what you'll note is that the sand is predominantly this very bright white color, which is typically indicative of being composed of a calcium carbonate mineral. And calcium carbonate in the ocean is typically made by organisms like corals or clams that make a calcium carbonate skeleton or shell. And if we were to collect a sediment sample from a beach like this, depending on where you are in the world, it might be composed of a bunch of different shells and skeletons. Here I'm showing you an example of a sediment sample from French Polynesia. And what you can see is there's an assortment of different skeletons and shells in this picture. We can see examples of gastropods like snails, foraminifera, coral fragments, crabs even, bryozoans, those are the sea fans that like to live in reef environments, bivalves, clams and mollusks, calcareous algae, and even urchins. You can see a picture of the spine on the upper left-hand corner of this photo. It has sort of like a pink, light and dark alternating stripes. That's an urchin. What you might not know is that fish are actually carbonate producers as well. So today we're going to be talking about their physiology and biogeochemistry. Here's our fish swimming in the ocean, participating in marine food web. Phytoplankton, those are the little green circles, are getting eaten by small fish, which are getting eaten by medium fish, eaten by our big fish. This big fish is also drinking seawater. As a process of drinking that seawater, it ingests calcium, magnesium, and carbonate. Those form a calcium magnesium carbonate precipitate, a carbonate mineral that gets excreted as ichthyocarbonates alongside fecal pellets into the sea. And these ichthyocarbonates are the focus of our research. My name is Dr. Amanda Ehlert. I'm an assistant professor in the Department of Marine Geosciences.
Hi, my name is Dr. Rachel Hoyer, and I'm a research assistant professor here.
My name is Eric Fulkers, and I'm a postdoctoral researcher.
My name is Martin Purcell. I'm a professor in marine biology and ecology.
And the four of us, along with research scientists and a team of undergrad researchers, are working together to address some important research questions about ichthyocarbonate. Our first question is, where does the carbon that forms ichthyocarbonate come from? We also want to know if big fish make big ichthyocarbonates, and we want to know what's the fate of ichthyocarbonates in the sea. And finally, we're really interested in the future role of ichthyocarbonates in the ocean. So in order to get started, let's talk in a little bit more depth about what are ichthyocarbonates and how they're formed as a byproduct of osmoregulation.
I'm going to talk to you about teleost fish and ichthyocarbonate, and actually also about osmoregulation. Teleost fish refers to a wide group of fishes. This is one of them. This is the gold toadfish, one of my favorites. They refer to fish that are bony fishes. They have spikes in their fins. They have spikes in, pretty much everywhere. They include tunas, mahi, toadfish like this one, tilapia, all the fish you typically eat. They do not include sharks and rays. They're not teleost fish. But by far, most of the fish in the ocean are teleosts. They're very good. And teleosts, they osmoregulate. And what osmoregulation means is that they maintain a certain salt and water concentration in their bodies. We do the same thing. And it's a problem for fish in salt water because salt water is very salty and very dehydrating. They're losing water all the time, just like you would at the beach when you're out in the sun and with the salt water. And they have to drink, and the only thing they can drink is salt water. And that's a problem. Don't try that at home. And part of the process of gaining water from salt water is that these fish form calcium carbonate precipitates inside the intestine. And those precipitates help by allowing for water absorption. And I'll show you one here, and I just picked that out of the tank. This is what they look like. These are calcium carbonates. And these are the ones that will end up in sediment and ultimately on beaches and form the white sand that you so like. So that is an ichthyocarbonate. It's involved in osmoregulation in these teleost fish.
One tool that is valuable for studying ichthyocarbonates is a type of microscopy called scanning electron microscopy, which allows us to zoom in and look at the ichthyocarbonate structure. This is what the microscope looks like on the left. And on the right, you can see an example of the types of images that we can get. For example, here we have a human hair. Let's take a closer look at ichthyocarbonates. This is an image I took from a toadfish after cutting the ichthyocarbonate in half. And this allowed me to see what the ichthyocarbonate looks like inside, and also allowed me to see that the ichthyocarbonate is surrounded by a layer of mucus on the outside. If we zoom in even further, we can see the individual ichthyocarbonates, both their size and their shape. One thing we have learned is that not all ichthyocarbonates look the same. On the left, we can see the ichthyocarbonate from the toadfish that I just showed you, while on the right, we see what mahi ichthyocarbonates look like. This is important because the size, shape, and type of ichthyocarbonate can change what happens to it after it leaves the fish. We can use a bunch of geochemical methods to look at things like the carbon isotope values of the carbonate. This can help us track the source of the carbon. So here on the left, I'm showing you the tools I use to pack the sediment samples, the ichthyocarbonate samples, into little tin boats that get put into a carousel. They get dropped into an acid reaction vessel where the CO2 that is produced from this reaction gets analyzed for its stable isotope composition on the mass spectrometer. It's orange on the bottom left, giving us an understanding of what is the composition of the carbonate part. We're also interested in any of the embedded organic matter and the organic carbon isotope values of ichthyоcarbonates as well. So in the middle section, I'm showing you a bunch of photos that show the steps of isolating ichthyocarbonate organic matter. First, I weigh them, I dissolve them in acid. We use a vacuum filtration setup to separate the organic matter from the carbonate. You can see in the middle on the left, that's a picture of some organic matter that was isolated from a sediment sample. I pack those in capsules and they get loaded onto another carousel on an elemental analyzer where they drop into a combustion furnace and turn into gas, where they can be analyzed for their isotope composition again on another mass spectrometer. And finally, mineralogy is a really interesting component of the composition of ichthyоcarbonates. And we take that little mortar and pestle like I'm showing on the upper right, and we powder the sample. We turn it into a paste and put it on a little glass slide and analyze it on an X-ray diffractometer, an XRD. And this can tell us what minerals are contained within the ichthyocarbonate precipitates. So let's dive into some of those research questions now that we understand how ichthyоcarbonates are made and what are some of the techniques we can use to study them. Each of us is going to go through and talk about aspects of the project that we're interested in to share with you. So first, we're going to start off with question number 1. Where does the carbon that's used to build ichthyocarbonate come from? So fish play many roles in the carbon cycle. They are a part of the biological pump. And the biological pump is an important mechanism that takes CO2 from Earth's atmosphere. It is drawn down into the surface waters of the ocean by photosynthetic organisms, and it passes through the food chain to various trophic levels where it either aggregates and sinks as fecal pellets or even phytoplankton down to the deep sea. And this is an important flux of carbon from the surface waters of the ocean down to the deep, where it can be sequestered or even stored for geologically relevant periods of time. And fish are part of the biological pump, right? Because they're part of this trophic transfer of energy and nutrients, and they also produce fecal pellets, which can sink to the deep sea, enhancing the flux of carbon from the surface ocean to the deep. But we also know through the production of ichthyоcarbonates that they participate in the carbonate pump. In normal marine organisms, when they calcify or precipitate their carbonate shells, they're going to pull the carbon from the seawater. And this, as a result, actually serves to release CO2 back to the atmosphere. Those calcite and aragonite shells or skeletons can sink to the deep sea, thus transporting alkalinity and carbonate to the deep ocean. Now, this is where it gets interesting because we know that fish are part of the biological pump, and we know that they produce ichthyocarbonate, which is a form of carbonate that sinks, so they're part of the carbonate pump. But what if there is a connection between these two processes? So by producing fecal pellets, they contribute to the biological pump, on the order of 1.5 plus or minus 1.2 petagrams of carbon per year. And they also contribute between 0.7 and 1.4 petagrams of calcium carbonate carbon per year. What if those two processes are connected? And ichthyocarbonate, since we know that a lot of the carbon that's in the ichthyocarbonate may come from their food, maybe there is a connection between the biological pump and the ichthyocarbonates that fish produce. And so we are using a bunch of those geochemical techniques I talked about earlier, using stable isotope mass balance analyses to track how much of the carbon comes from diet and how much of the carbon comes from the seawater. And the ratio of these two carbon sources is going to be really important for understanding the role that fish play in the global carbon cycle through time. Our second question is, what is the fate of this ichthyocarbonate in the sea?
So in addition to the uncertainty about how much is being produced in the surface waters in the oceans of this ichthyocarbonate, we know very little about where this ichthyocarbonate is going. There are some studies out there reporting ichthyocarbonate in the sediment, but it's the sand at the bottom of the ocean. Those studies are from shallow water environment, and there's no disputing it. Some of these ichthyocarbanates do reach the sediment. But from sort of a global or whole ocean perspective, we really don't know. And ultimately, it's going to come down to two factors. One is how soluble these carbonates are. How likely are they to dissolve in water after they're released from the fish? And two is how long are they going to be in the water column before they reach the bottom and get scurried or integrated into the sediment? Those are the two factors that are going to matter. We don't know a lot about it. We've just started working on it. What we've done so far on the Gulf toadfish is we've measured the dissolution rate. And our measurements show that ichthyocarbanates dissolve very fast, much faster than any other biologically produced carbonate in the ocean. So that suggests that they're not perhaps as likely to reach the bottom of the sediments as some other carbonates. But we have to admit that toadfish likely have some of the higher dissolution rates because of the chemistry of the ichthyocarbanates. It differs among species, and we have reason to believe that toadfish are going to be among the faster ones. So we need to do these measurements in other species. The other factor, sinking rate, is a product of two things: the size of the ichthyocarbanates and the specific gravity, so their weight. And they are heavier than water, so they do sink. But that sinking rate is going to be very much influenced by the size. And so we're working more on that to kind of determine just how long are they going to be in the water column, and is that time going to be sufficient for them to fully dissolve, or is it going to use some of the precipitates to reach the sediment?
So we want to know, do bigger fish make bigger ichthyocarbonates?
I am basically looking at kind of the interplay between the sizes of fish and how that might be impacting the size of carbonates that are being produced. And it's an important kind of thing to consider because fish, you might think a bigger fish just creates a bigger carbonate, and a smaller fish creates a smaller carbonate. And it might be a linear effect, it might not be. But it's important to know because the size of carbonate will significantly dictate and impact where that carbonate travels and kind of goes to the water column, and if it's able to reach the bottom of the ocean and settle. And we have preliminary data that shows just this, that size of carbonate is a huge factor in determining some of those things. And it's important to know because when you think about in the world's oceans, the vast majority of fish are in and around that 1 gram size. So these pretty small fish. And we kind of need to know these smaller fish, are they creating smaller carbonates or different size carbonates? And are we able to predict just on fish size alone to understand what size of carbonates might be being produced? And that's kind of important too, because once you kind of get an idea of that, we're then able to kind of go through and model potentially, because we know where there are worldwide certain fish biomasses at different sizes occurring. So if we know that, we kind of get an idea of the sizes of carbonates that are produced in those areas, and therefore kind of getting an idea of where there are carbonate production hotspots occurring and what those sediment potentials are of those carbonates which are being produced by those fish at those different sizes. So they all kind of interplay together, kind of get a better idea of what's going on with the carbon cycle and about the different sizes of fish, what sizes of carbonates are being produced, and how that might be impacting sedimentation.
And last but not least, Rachel's going to tell you about the future role of ichthyocarbonates and why we are interested in their participation in the global carbon cycle.
One future direction we are currently exploring is how future projected climate change could alter ichthyocarbonate production. Increases in atmospheric CO2 will be mirrored in the ocean, leading to rapid and widespread ocean acidification. As waters become more acidic, fish change their internal body chemistry in a way that makes them produce even more carbonates. This is important because areas with many fish that are supplying carbonates could help other organisms that need carbonates to build their skeletons, like corals. This research will also tell us more about how the global carbon cycle will look in the future.
Well, thank you all for your attention. We are happy to take questions. I see a few are popping up in the Q&A. Oh, good question. What are the black fecal pellets and the white fecal pellets? And why are the fecal pellets not the same color? So the fecal pellets are made of organic matter, and so they usually end up looking pretty dark. But the ichthyocarbonates are made out of that calcium carbonate mineral, calcium magnesium carbonate mineral. And so they have a very white color, similar to the white sand beaches we were saying, talking about in the beginning. So because they are made of two different types of carbon in different phases, they often have very different colors. That's a great question.
Wonderful. Thanks, Dr. Ehlert. You got to the questions in the Q&A box before I even had to mention it. I want to mention to all of our participants to please put your questions into that Q&A box. All of the presenters will be staying on for several minutes following their presentation. They'll be able to take those questions, if not live, but then afterwards in the chat. So great question, Jelena. And we'll give it another minute. Otherwise, folks, if something comes to you, please throw it in that Q&A box. And I thought that was a really fascinating overview in a field of study I truly wasn't familiar with. And it seems like a lot of really intricate technology is involved.
Yeah, thank you. It's really interesting too. As a geologist, I never thought about fish precipitating sediments or minerals, and meeting Rachel and Martin and Eric has been so fun to kind of look at science from their perspective and see how geochemistry can support some of our research questions. I don't know, Rachel, if you want to add anything as well, feel free to chime in.
Yeah, it's been a lot of times we sometimes focus in our own, subject area, and I would mirror what Amanda says. It's been really fun to kind of take two different fields that normally maybe wouldn't interact very much and put it together and then come up with some really cool research questions that we're really looking forward to exploring further. It's been really fun.
And that's right there. The definition of science is this constant pursuit of answers. And this was a really fantastic way to open the event. So thank you both for being here. And folks, they'll be available to engage with you for the next, I don't know however long they're willing to stay on. So thank you very much, Dr. Hoyer and Dr. Ehlert.
Thanks for having us.
Thank you. Thanks for joining, everyone.
Yeah, we're gonna transition now from the world of bony fish to that of marine mammals. And we're gonna welcome Dr. Maria Cartolano and Dr. Jill Richardson and a group of their students who are gonna be speaking about marine mammal science here at the Rosenstiel School. See, we have a screen share. All right.
All right, thank you guys so much for being here. My name is Dr. Maria Cardolano, and myself, along with Dr. Jill Richardson, are the coordinators of the Marine Mammal Science Lab here at Rasmus. Today, you're going to hear from a couple of our graduate students who are in the Marine Mammal Science track in the Masters of Professional Science program here at Rasmus. So they are in this program to get hands-on experience through classes and experiences to become marine mammal scientists and conservationists. So they're getting lots of different experiences, and they're going to share with you today the careers that they are pursuing, and then some of the career options that are open to you if you are interested in joining this field. They're also going to share a couple of their personal experiences with diversity, inclusion, and accessibility in this field. Just to give you a quick introduction into what marine mammals are, we classify them into 4 different groups ranging from fully aquatic to semi-aquatic. And so we have our cetaceans, which include our dolphins, whales, and porpoises, our pinnipeds, which are our seals, sea lions, and walruses, our sirenians, which are our manatees and dugongs, and then the marine fisipeds, who are polar bears and sea otters. All right, next I'm going to pass the mic to Madeline, who's going to talk about marine mammal veterinary science.
most, but I do a lot. I'ma make a toast, cause we still alive. Nope.
Hi, welcome. What up? I'm Natalie, and we're in the Marine Mammal Science Department. I come from the Memphis 10901. That's what I'm representing, the city of Young Dolph, Young Gotti, Yo Gotti. So this is what we're going to be talking about. We're going to be talking about careers in marine mammal veterinarian. And I know you're like, marine mammals have veterinarians? Yes, dolphins, whales, seals, manatees. They have to go to the doctor, just like your cat, your dog, your lizard, bird, whatever you have. So these are just a few pictures of what marine mammal medicine looks like. As you can see, the different procedures that are going on in the pictures. Okay, so excuse me, I'm sorry, let's go back. So as a marine mammal veterinarian, most of your animals are in the water, as you can see. So which makes it kind of difficult to access them to do those medical procedures. So what they do mostly is they'll get your blood like they do with the cats and dogs. They'll get your fecal, they'll do x-rays, ultrasounds, and all those things that they need to diagnose the animal to make sure that they're healthy. And then as a veterinarian, you can work in different fields such as the government, facilities, Georgia Aquarium, SeaWorld, or you can do stranding networks. And then if you're not the person that wants to touch the animal, but you want to work behind the scenes, then you can be like a researcher, PhD veterinarian, and do the pathology. That's a study of diseases, looking at their tissues on slides. That's histology. You can look at the toxins in their body. That's toxicology. So those issues, you have different ways to able to access marine mammals as a veterinarian. Okay, now let's talk about carving your melanated space in marine mammal science. Most of the time when you look at marine mammal science, where this is why I'm going to keep it real for you, 100, you don't see a lot of melanated people. Usually in the veterinarian field period, it's less than 2% African Americans. So that means it's going to be less for the Hispanic community, the Asian community, and the indigenous community. So it's not a lot of representation, but that's okay because we're coming, okay? So then, for example, it's only 2 Black veterinarians, females in the United States, 1 Black male veterinarian, and he just left the United States to go open up and head vet at SeaWorld in Dubai. And then there's 1 Black Trinidad marine mammal veterinarian. So that shows you how people of color are not really in the field. And I can count on my hands how many researchers are actually out there doing the research, marine mammal trainers, the people that you see at SeaWorld helping you show their natural behaviors. So what to expect as a person of color in this field is some things that you should expect or know that might happen. You may be the only one, which is okay. You can expect some people not to really take to you. They might feel hesitant about interacting with you. They may give you like the side eye or something, or, things that just is not, could be discouraging, but don't worry about that because you are there and you're there to just be you and do you. So and then sometimes people might tell you, I don't like your vision, but that's your vision. So, and you continue to pursue your vision. So you don't let anything discourage you. You be prepared. You keep support around you. You continue to be your authentic self, and you just keep moving forward. And you don't let nobody take anything from you. So what do you need to do? How do you handle these situations? You're going to show up. That's what you're going to do because you deserve to be there, okay? Remember, you've earned your right to be there and you've worked your butt off. Do you hear me? All that studying, all that hard grind, and all that knowledge. And then you find you a safe space. Have your classmates, faculty. And then next, encourage yourself. You have to always encourage yourself because you are the game changer. And that's who you are. You occupy that space. And what I do as a game changer, I have to have that instant Ray moment when you talk to yourself in the mirror and I say, I am Dr. Natalie Jones. and you will know my name in this field. So with that being said, I'm here to lay that groundwork for you to come to Rasmus, because Rasmus is a great place. And I hope to see you, and I hope you will come with me to help improve diversity. And I'm out.
Hi, everyone. I'm Megan. I'm going to be talking to you today about marine mammal research, since that's my field of interest. So some major threats to marine mammals, they can both be natural and human caused, like pollution and contaminants through either there's plastics and microplastics. Commonly, plastics are found in dolphin stomachs. It's one of the most common things found in a necropsy. Entanglement fishing gear, boat strikes, their prey are commonly overfished, harmful algal blooms, disease, and then harassing and illegal hunting. Noise pollution will be touched on later in the presentation. Most of marine mammal research is centered around understanding marine mammal behavior, biology, ecology, and finding ways to protect them. So the more we know about what's harmful to their populations, the more we're able to help them. So some hot topics in marine mammal research right now are the topics of genetics. So marine mammal scientists can learn a lot from DNA analysis, studying maternity, social hierarchy, and how different groups interact, as well as discovering new species and new classifications through DNA analysis and evolution. It's really important also to understand physiology, so we understand how their bodies work. So, scientists are able to understand how they're able to dive miles down below the surface, how they respond to stress, how their sensory systems interact with others. And the more we know about this, the more we can know when something goes wrong in their body. And just like Natalie said, we need to do more research to understand how to medically treat mammals. For example, there's a wild killer whale off the coast of the western coast of the US named J50. And because of overfishing in the area, a lot of J50's food supply was depleted, the Chinook salmon. So a team of qualified biologists, officials, they all stepped in with all their knowledge and experience from the research, and they actually provided medicine to her through a dart. And that was very revolutionary to get her the nutrients she needed. Researchers will also investigate how pollutants and everything affect marine mammal health, such as via the Deepwater Horizon spill. POPs, or persistent organic pollutants, are what build up in the marine mammal's blubber layer, and they can be very harmful, as well as plastics and microplastics in the stomach. Scientists also will look into the ecology and distribution of marine mammals to know where they're migrating, how many there are, and where they are going. So some other topics, so like wild research for marine mammals, it's very difficult to do because they are underwater. So photo ID surveys through aerial and boat-based drone surveys are very interesting and a good way to develop population estimates and evaluate social behavior. A lot of this work is done with photo ID and cataloging different populations for different species. Like for dolphins, as you can see on the right in the pictures, their fin is like, their dorsal fin's like a fingerprint to them. So it's unique, and that's how scientists are able to track them over time, by seeing that dorsal fin. For marine mammal health and toxicology, we do these things called HERE studies, which are health and environmental risk assessments, which involve capturing and conducting extensive medical evaluations of wild animals before releasing them in at-risk populations. These efforts require extensive permitting and expertise, and they tell us a lot about the ocean environment and dolphins as sentinel species. Scientists also collect non-invasive biopsies from their blubber layers so they can evaluate for toxins and biotoxins and pathology, as Natalie talked about. And tagging with sensors helps them get information about how they use their habitat, diving, feeding, and how long and how deep they're diving. So something else, another way that we can evaluate them outside of the wild is through the lab. So in lab experience, we're limited via necropsy or model organisms due to ethics and animal research laws. So necropsy is the examination of dead marine mammals, and usually from a stranding event. So it gives a lot of information on marine mammal physiology and overall health. And in the lab, scientists can also analyze samples from wild marine organisms. Marine mammals that are commonly in managed care can provide a lot of critical information about biology and behavior that can be used and applied to wild populations, and really helpful in research and conservation. These animals are also trained voluntarily to participate in these research studies, so you can learn about their social and reproductive behaviors, how they communicate with sound, growth, and how we can treat different diseases. So something I've personally participated in was doing behavioral ethograms at Dolphins Plus Marine Mammal Responder in Key Largo. And what you would do is every 60 seconds, you would have a timer, you look up, you record exactly what that dolphin is doing at 60 seconds with only your focal dolphin, who it's with, its proximity, its exact movements. And then you just look down until the next 60 seconds. And we do that for the first year of life to get an idea on their behavior and social growth. Overall, studying marine mammals is extremely helpful to understand, and managed care is very helpful to help understand how they protect their wild counterparts. And without managed care, we wouldn't be able to understand those populations as well as we do now.
Hi, my name is Danny Barrios. Thanks. I'm going to talk a little bit about my experience in diversity in the marine mammal field. So my parents are both from Cuba, but they met in Mexico, and my dad went to school in the Dominican Republic. So I'm pretty connected to that part of the world. However, when I was young, after moving away from Miami shores, where I spent the first 5 years or so of my life, I lived in a place where I didn't have a lot of that experience, and it made me feel very disconnected from that world. So especially nowadays, I find myself trying to incorporate as much of it as I can in my current self, especially being able to be here in Miami, where there's a large Latino population. Yeah, so I just try to bring it out in myself, and I just try to be who I am, because it's important to bring every part of your heritage and every part of what you want yourself to be. Nobody can really decide who you are except yourself. So it's important to keep that in mind. And yeah, with that, I'm going to talk a little bit about marine mammal acoustics. So we use sound in a lot of different ways to study marine mammals. For one thing, we can put hydrophones, which are these underwater microphones, in the water to get some of their sound. And with that, we can kind of look at their habitat use, we can look at their abundance, we can look at their migration patterns. So this is really important because marine mammals use tons of sound to communicate with each other. And we'll go into that more in a minute. We can also use these sounds that we've recorded and put them into computer programs to analyze how they work. So you can see in this spectrogram, which is this like green box at the bottom, you can see the way the stripes that are going through it. Those are sounds of marine mammals. We can also look at their social behavior, because these animals have tons of different types of calls, including signature whistles for dolphins, which is kind of like them calling out to each other like a name. It's like this unique and distinctive tone that they can all use to recognize each other. So yeah. Okay, so for marine mammal acoustics, obviously, like I mentioned, sound is very important to them, and it's a very important part in their daily life. They use it for navigation. They kind of use it as like a GPS system. And they can move themselves in space using this navigation, using sound. They can use it to find prey, like dolphins use echolocation, which I'm sure most people have heard of, to bounce sound off of objects. See if we can get the sound to play. Yeah, those are some noises that dolphins can use to locate the prey. And they can also use it for communication. So humpback whales have very long and complex songs that they can use to communicate to each other and just to kind of orient themselves a little bit. So we can hear some of these. As you can see, it kind of sounds like a creepy choir of sorts. And they can also use it to avoid predators, which is very important to be able to get away from things. So cetaceans in the sounds that they use, they're super developed. They tend to stick to high frequency, especially cetaceans such as dolphins and porpoises. But when you start looking at other cetaceans like whales, they tend to keep to low frequency sounds that are longer, and they can even have different accents, which are different patterns of clicks that these animals can use to communicate. And yeah, I'll move it on over to Lindsay to keep talking about sound.
All right. Hi, everybody. My name is Lindsay, and I'm going to be talking a little bit more about noise pollution and how that affects marine mammals. So first off, sound propagation is 4 times faster in water than it is in air. So sound travels extremely fast. and long distances. Anthropogenic sound is man-made noise, and that affects marine mammals in various different ways, in which I'll cover in a little bit. But some of the biggest culprits I have highlighted here, and the top 3 are going to be the ones in bold. So commercial shipping for shipping goods, seismic exploration, which is imaging oil and gas. And I have a couple pictures here that you can reference, as well as sonar. And that's basically mapping of the Earth's crust, and a couple others, explosions, acoustic deterrent devices, which are actually put on fishing nets in order to keep marine mammals away from the nets, so getting caught in them or taking the fisherman's catch, and then other things such as personal watercraft and drilling. A couple sounds to listen to. So if you could tell, it was really hard to hear the sound of the animals in the background of all of that anthropogenic noise. So just imagine what it's like for a marine mammal traveling underwater, trying to do all of those activities and important things that Danny talked about, and not being able to do so because of all of the loud noises that are happening. So the effects of noise pollution are wide ranging, and there are various different outcomes. So first of all, physiology and behavior is altered. They can lose their hearing, they tend to avoid, as well as habituation occurs, which is the hearing of noise repeatedly and getting less sensitive or getting desensitized to it, and also causing stress. Just like us, when we're stressed out, we obviously have a really hard time doing regular things. Masking occurs as well, so they have a harder time hearing their family members. Strandings is a really big one, especially for beaked whales, and then growth and reproduction is also altered. And how do they overcome the noise pollution? Many different ways. Louder calls, they increase the amount of calls that they're doing, they repeat it, they increase their frequency to maybe make sounds outside of the frequency of the noises around them so that they can be heard. And also they increase for the duration of time that they make their sounds. All of these result in a decrease in fitness, in which I had on the last slide, and it's extremely energetically expensive for these animals.
Hi everyone, I'm Mackenzie, and I'm going to be talking about rehabilitation and strandings with my partner. And I'm from Texas, and a lot of my work has been mainly with strandings for sea turtles.
Hi, I'm Jory, and I'm from Nashville, Tennessee, and I'm really passionate about marine mammal strandings and rehabilitation, which is what we're going to talk about now.
So what is a stranding? A stranding occurs when a marine mammal is found dead, either on the beach or floating in the water, and they can also be alive on the beach or unable to return to the water. And so some common causes of marine mammal strandings are noise pollution, like Danny and Lindsay just talked about, disease, which can spread in between marine mammal populations, entanglements, which typically do occur from anthropogenic interactions, harmful algal blooms, which can affect their respiratory and digestive systems, accidental beaching, which can occur when like the geography is weird or if the tide is going out and they're at the wrong place at the wrong time. But sadly, a lot of the causes of strandings are unknown.
So once the stranding response team arrives, if the animal is already deceased, then they can perform a necropsy to determine the cause of death. Or if the animal is alive but very ill, it might be euthanized on site. Or if the animal is just entangled or accidentally beached with no other illnesses or injuries, it might be released back into the ocean, but this does not commonly occur. And in other cases, the animal may be transported to a nearby rehabilitation facility where it can receive further care and assessments so that it can be released back into the ocean once it's healthy again. So of course, we don't want marine mammals to strand, but when they do strand, it provides really unique and valuable opportunities for us to learn more about them and their environment. So from strandings, we can learn about life history, such as survival, mortality, and reproductive maturity. And we can also learn how diseases and infections affect their health. And we can also do population assessments because, for example, if multiple animals strand together, we might be able to learn more about social bonds And we can use photo ID to learn more about that particular animal, and we can confirm whether it's still alive or deceased. And we can also learn about environmental conditions because algal blooms, chemicals, and oil spills can cause illnesses in these animals, which may cause an animal to strand. And we can also assess fishery interactions with marine mammals based on fishing gear entanglements and injuries, because these might indicate that regulations may need to be implemented to protect these animals.
And so within strandings, there are a lot of different career options. So there's different stranding networks all along the coastline throughout the United States. And each of these does have a stranding coordinator. So this is kind of the head of the program for that region. And within these programs, there's also a stranding biologist and a research technician. So these are the people who are typically performing the necropsies, which I think Megan and Natalie told you guys earlier is pretty much an autopsy on an animal to help determine the cause of death. And so the biologist and technician typically are the ones who are collecting and processing samples and data and trying to analyze the cause of death. And then when there is a live stranding, veterinarians are also super important because they are the ones who will assess the situation and decide whether the animal should be brought in for rehab, re-released, or in some cases euthanized because it's in the best interest. But the best advice we have for anyone wanting to get into strandings is to get in there and volunteer and intern early because they always do need more help.
So once a stranded animal arrives at the rehabilitation center, it undergoes multiple medical exams, such as those to assess body condition, because typically these animals may be malnourished and underweight. And then we can collect biological samples such as urine and blood, do x-rays, use ultrasounds and other diagnostic tools to determine what needs to be treated. And it's really important that these animals receive proper nourishment and hydration and any necessary medicine so they can heal and begin to build their strength up again, so they can eventually be released back into the wild once they are healthy. And as long as environmental conditions are right, because for example, you wouldn't want to release a rehabilitated and healthy animal back into the ocean if there's currently a red tide bloom. However, some animals are deemed unreleasable, and this is typically because they may not be able to find food on their own, or if they were rescued at a very young age, they may not be able to form those important social bonds necessary to survive in the wild. And so in those cases, those animals stay at the rehabilitation facility where they receive continuous care. But it is the ultimate goal of all rehabilitations to release those healthy animals back into the wild.
And so we did want to give a more specific example to a current issue that's happening in Florida, which is the Florida manatee. So lately we've been seeing a lot of these animals coming into rehabilitation facilities, and this is mainly due to their susceptibility to cold water temperatures. And so during the winter months, they'll congregate usually in warm water sites in the springs, and this is where they'll feed during the winter months. However, lately these harmful algal blooms happening in the state are causing a lot of the seagrass to die, and this is their main diet. And so these animals are coming in super emaciated, usually from starvation. They're very malnourished, and these boat strikes can also happen as well in these areas, and it'll cause pneumothorax, which is air in their chest cavity. And so these starving and injured manatees are rescued and taken into rehabilitation centers such as Miami Seaquarium and SeaWorld, where they'll receive nourishment and care, and then they'll be released back into the wild once they're healthy. And so you can kind of see this bottom picture. A bunch of us within the marine mammal program got to help release one of these manatees that was rehabilitated back into the wild, which was super awesome and cool to see.
Hi guys, my name's Cameron, and I'm going to talk to you guys about accessibility in marine science and just kind of my experience going through this field and through my undergrad experience. So I was born with pretty severe hip dysplasia and Perthes disease, which means that my hip is pretty malformed and it kind of looks like a rock compared to my other hip. It required a few surgeries and body casts as a kid, as you can see in my pictures. But I had a pretty good outlook. My doctors didn't really think anything would happen, but unfortunately they were wrong. And when I got to high school, I started experiencing a lot of complications from these issues. I started having really bad back spasms to the point where I would have to stay home because they were so painful. I had leg pain up and down most days, and I just had sciatica, and it was just a very painful experience for me. And at the time, I mean, I was 15, 16 when this was starting. So I was pretty confused and I didn't feel like a lot of people were there to help me out. And my doctors weren't really listening to me. But when I got into college, it unfortunately got a lot worse because I was walking around with a big backpack and it got to the point where I couldn't walk more than 10 minutes at a time. So I was not having a very good undergraduate experience, unfortunately. And around this time, I had been bounced around to like 5 or 6 different specialists, and none of them could really fix what was wrong with me, and no one could figure out why I was having these issues. But fortunately, I found a doctor and I underwent a leg reconstruction surgery between my sophomore and junior year of college. They went in, they broke my femur and they twisted it because it was twisted the wrong way. And then they went up, they dislocated my hip and shaved down the bone spurs. And then they reattached a muscle that had been flapping around, and they broke and re-angled part of my pelvis. And they put me all back together with a bunch of screws. And I returned to college a month and a half after that, a week after I was given the all clear to put pressure on my leg. And that's when I really started noticing some issues on the college campus. So even before the surgery, I had a lot of issues getting to class in general and not having the same 24 hours in a day as other people. Because when I would get home from classes, I would be exhausted, I would be in a lot of pain, and I would just want to sleep. Compared to my friends who would want to go out, maybe go to a party, go to a game, or just do homework and study, I didn't have the same 24 hours. And it was really hard for me. I'm not going to lie, it was a struggle. And I think a lot of people in my position are kind of conditioned to suck it up and not really make a fuss about it. And a lot of the professors and their syllabi don't really reflect the chronic variability of someone. But even though it was a really hard experience for me, I don't regret doing it, because the more people that are in this field that look like you and me, the more change can happen. And so if you are going to go into this field, my advice is to just advocate for yourself, because the more you advocate for yourself, the more people are going to listen to you. And if people get mad at you because you're advocating for yourself, those aren't people you want in your life anyways. And then creating a support system was really what got me through undergrad. I had a support system of friends, professors, even like a family that lived nearby, and they would always drive me around. They would do what they can. And just finding those people that will answer the phone when you call is super important. And just taking care of yourself, because your grades are always going to be more important, not your grades. Your health is always going to be more important than your grades. No matter how important you think that paper is, your health and your mental health are the most important. And so I have this little clip, I'll only play like 10 seconds, but this was me during my undergrad, the day after I had my surgery. I was trying to walk. And I had to relearn how to walk during my college experience, but here I am 2 years later, and this was a class where I got to work with dolphins, and I'm in my dream field. So it does get better. And if you stick through it and you support yourself, you find your support system and you advocate for yourself, it gets better.
So we just wanted to thank you all for tuning in. As you can see, there's lots of different career options in marine mammal science. You can go into training, you can go into rehabilitation, strandings, veterinary science, conservation. You know, there's lots of jobs opportunities for you. And I hope that we're inspiring you to go into this field, and we want you to know that you're welcome in our field. Thank you so much for attending, and we appreciate your time.
Thank you, Dr. Cartellano. That was super interesting, and what an interesting way to open and close the scientific content. I mean, and I think there's something to be said that no matter what field we go into, while the focus of this particular event is marine and atmospheric science, starting out with the message of advocating for oneself and being your own cheerleader and using your voice and speaking out, that's something that we can all carry forward throughout all walks of life. So it was just really wonderful messaging from your students, in addition to the super cool information that they presented. There were a couple of questions. I think people are getting warmed up with the Q&A box, and I'm going to ask maybe Dr. Cartellano if you can answer one that came up in the chat, and it was from one of our viewers, Catherine, who asked what the average age of a PhD professor at Erasmus is. So if you could key us in on that, that would be great.
Yeah, that's a really good question. I don't know what the average is by the time you finish your PhD, and then you kind of usually do a postdoc and get into your career. I would say the youngest you could probably become a professor is around 30, and then you can stay in it as long as you want. So I don't know what the average is in our department, but you can definitely get in there young, or you can stay in it as long as you want.
Wonderful, thank you. And for all you folks watching from home or wherever you are, please feel free to stay on and engage with our presenters in the Q&A box. Next, I have the pleasure of introducing Dr. Vayed Shirayath and his student Stephanie Wright, and they are going to be speaking to us about inventing NASA technologies to help map the ocean.
Thank you very much, and a pleasure to be here. Everyone can hear me all right?
You are
Okay.
great.
I'm going to try sharing my screen now. How does that work? Does that come through?
Yep, we see it.
Okay, so my name is Professor Vayed Shirayath, and I'm here with Stephanie Wright, who's actually just in the office next door on the weekend at Erasmus. That's how motivated we are to be here. I'll talk a little bit about some of the technologies I invented at NASA. I directed a center there for 10 years in Silicon Valley before moving to Erasmus just late last fall. And I'm continuing to develop these technologies here with NASA support at the University of Miami. And I'll share some of the new technologies we're focusing on for oceans across the solar system, which may be a little strange of a title to you. You think, okay, there's only Earth's ocean. What are the other ones? I'll get to that at the very end, but we are not alone in having an ocean in our solar system. And it's a very exciting time to be alive, to get to explore what is perhaps the least known thing on our planet, and certainly in our solar system. So, diversity for me is really key to just mission success. And you'll see this ethos shared not just at NASA, at the University of Miami, but across science. Truly the best science comes from, having a diverse set of people that bring those ideas to the table. I was listening to some of the other speakers and I felt, these issues are real and they're continuing. As someone who identifies as LGBT myself, we're very few and far between in my field. In fact, I think my last center, I was the only one. And there's a lot of, change that is happening, but you do have to kind of be that example and that change to see what you want implemented. So this is just a snapshot of my NASA team. Stephanie, unfortunately, isn't in this one yet. She'll be at the next one. But this was one of our field campaigns in Guam. And it's just an example of, what kind of great team building can lead to good science. So, all right, what do I do? My background is in astrophysics and theoretical physics, and then I moved over into ocean science and trying to understand Earth. And the main reason was this fact, which is still true as of 2022. We have mapped more of the surface of Mars, the sun, the moon, than we have of our own ocean floor. And I mean, we've literally mapped all of that. Picture of the moon I took. Mars is from a NASA satellite. The picture of the sun I took, we can see all of these surfaces because they're not occulted by water. On Earth, 70% of our surface is water, and most of our planet and most of its life is underneath that surface. So coming from a space agency where we're trying to look for life elsewhere in the universe, trying to understand it as best we can on Earth, we really have to be able to understand it on Earth to extend that search elsewhere. So why is it so hard studying things in the water? Well, this is a little bit of physics for your Saturday morning. We have this problem of attenuation, or what we call absorption in the water. So water absorbs light, right? If you look out at the ocean, it looks blue for various reasons, but mostly it's blue because it's absorbing everything else that's the sun is shining down upon it. So in our atmosphere, we have a few windows that we can use to look up at stars in the night sky. You can see the visible light, you can see visible light from stars. You can also see radio really well through the atmosphere, but in water, all of those get absorbed in the first micron of water, or the first tiny layer of water. So you're just left with visible light. And really in that visible light, the blue kind of gets the deepest. But if you're down to 100 meters depth and you look up, it's essentially dark. And the average depth of the ocean is 4,000 meters. It's not 100 meters. So this is the main problem with trying to study anything from space or from air. It's very hard to see something underwater. The second problem is refraction. Now, this is something you learn about hopefully in elementary school or middle school. You have a fluid like water and air meet, and they have two different indexes of refraction, which means that light gets bent. If you put a pencil in a glass of water, it will look like it's crooked, but in fact, the pencil is whole, right? The same phenomena happens in the ocean with ocean waves. And the problem is that when we try to look at the ocean with our NASA satellites or our aircraft or drones, that those waves distort the view. They really inhibit what we can see. So at the bottom left, you can see what a coral reef looks like under these different wave conditions and from different satellite imaging systems. So right now on land, you can pull up almost, for most parts of the world, an image that is 1 meter resolution or so. That's mostly coming from NASA data or from commercial satellites. In the water, you cannot do that, right? You can't go on the boat and just pull up a map and say, oh, here's a piece of coral that's at depth and see it with that resolution. So this is one of the main challenges we have with imaging things. And so the focus of my lab and my whole career is developing technologies that can get through that barrier. Two of those are called FluidCam and MIDAR. I won't talk about MIDAR today. I'll focus mostly on FluidCam and the fluid lensing camera. And then the third technology is, what do you do once you have mapped this information? How do you interpret it meaningfully? And this is actually a video game development that's tied to a supercomputer called NASA NemoNet, and I'll share that at the end. So what is FluidCam? This is an instrument we built into a small CubeSat platform. These are small satellites we can put up into orbit cheaply. Right now, this satellite actually flies around trying to map corals on aircraft. And this is the problem. So if you look at an aircraft picture of something underwater and you have a high-speed camera, you can see that middle picture at the top. You see the waves distorting the coral. That's the problem that we're trying to fix. Fluid lensing and FluidCam can create the picture at the bottom right. So what it does is it tracks all of the times a wave magnifies the object. So in this case, a piece of coral, the wave will pass over it and you can momentarily see it kind of act like a little magnifying glass, right? The instrument's able to use that like a free telescope to see at very high resolution. And then the second phenomena that FluidCam uses are those bright bands of light that you see dancing on the seafloor at the bottom of a swimming pool. That's actually coming from these lenses, these waves on the surface of the water, focusing sunlight down on the bottom, and they're called caustics. These caustics can actually be really, really bright, so you shouldn't look up if you're swimming underwater at the sun. But what's nice is from our instrument, we're able to track those caustics to see very, very deep in the water. So by very deep, I mean still within the first 100 meters of water, but it's a much brighter picture than we would get if we were to flatten the ocean surface like we see on the bottom left, or if we were to drain the ocean in the top left to get that view that we would normally get on land. So we fly this instrument right now around the world trying to map coral reefs and understand their 3D structure. You may ask why corals? Well, the latest COVID treatment came out of a coral reef. So that's one of the reasons why they're the 21st century cabinet of medicines, because these creatures are very advanced in terms of evolution. You saw right there what the view looks like before and after fluid lensing. So I can try to skip ahead again here. So from the aircraft, you see this distorted view with the reflection of the water. And then after fluid lensing, we get this 3D view underwater. And it's essentially a diver scale view of that whole reef at scale. So we can map tens of square kilometers at sub-centimeter resolution, which is a huge amount of data. Most recently, this instrument was able to get much deeper in the water column. So this is from a field mission in Guam, where we're actually mapping things at 45-foot depth in 3D at around the centimeter scale. So you can see examples of what some of the corals look like at that depth. But this is kind of the first time we've ever been able to see that deep in the water without having to get in the water. This is an example of that same data set compared to a satellite picture on the far left. So it shows you kind of the improvement we're trying to get with an aircraft. And then eventually we're going to try to put this up into space. So we have a fluid lensing satellite that can do this. And we can also pick out now with the new technology, wildlife. So, things like sharks, fish in the ecosystem. In addition to mapping the seafloor, we're also trying to understand what is between the seafloor and the surface, because that often tells you a lot about, for example, the health of coral reefs in the case of sharks. If we see sharks, it usually means that a reef is quite healthy, not always, but usually. And here's an example of some of the new platforms we're putting this instrument on. So, between drones and satellites, there's a whole bunch of other platforms. We've been developing a solar powered high altitude drone that's quite large. You can see the wingspan there, it's about 80 feet. This flies for 3 months at a time, and it can hover over islands. It doesn't use any fuel. It's completely solar powered. And the idea would be that we can, for marine imaging, it's a much slower process than trying to do land imaging. So it takes quite a bit of time. And the idea is we could fly this over different island locations and slowly build up a picture of what's on our planet and what's underwater. So we've been doing field missions, again, pretty distributed around the world. We're going this summer. Stephanie's helping coordinate a field campaign to Hawaii, Guam, and Palau, where we'll be knocking out more mapping of corals. And the question now you might be asking is, okay, you've been mapping all of this. What do you do with all that data? So just imagine the amount of data we have for land is around 10 petabytes per year of satellite data. Petabyte is 1,000 terabytes. 1 terabyte's about 1 laptop. So it's a huge amount of data. And now when you map the ocean, it goes up exponentially, right? Because it's a much bigger surface. So we're actually asking people like you, citizen scientists, to help get involved and train our supercomputer to classify the coral reefs by itself by you painting on it. So I'll share a short video of how this works.
What if you could help NASA create a map of the ocean floor with just the tip of your finger? The ocean, teeming with life. It defines our blue planet, drives our ecosystem, and regulates our climate. Coral reefs are one of the most diverse and important systems in the ocean. They're also becoming an important source of medicines for some of the world's deadliest diseases. But they are dying at unprecedented rates due to rising temperatures. But we don't know how much we're losing or how much our climate is changing. We can't, until we determine how much healthy reef exists now. And the only way we can know that is with your help. NASA NemoNet is a game where you classify the world's coral reefs by painting on real-life images scanned from the ocean floor using a revolutionary instrument that lets us see beneath the waves at unprecedented resolutions. Our oceans are so vast, it would take us 2 million years to classify the world's coral reefs by hand. The classifications you create are sent to our teams of NASA scientists at home base to teach our supercomputer to classify coral reefs on a global scale. Every contribution you make brings us closer to solving this problem. Join the NASA team to help us understand these amazing ecosystems. Take command of your research vessel and learn about all the different types of coral. We must keep the ocean alive. It supports our life as we know it. Together, we can create a global data set of coral reefs and build a better understanding of how to save these aquatic worlds, one piece of coral at a time. Good luck, and welcome to the NASA NemoNet team.
So you could play this now on many devices. It became very popular in the peak of the pandemic. We had about 300 million people just in the first month accessing it from all over the planet. And we've now been translating it into different languages to try to make it more accessible. Here's just some of the, shows you kind of the spread of the technology. And so it's not just in island nations that have coral reefs, people who are landlocked or, interested in mapping corals, and they're producing really good scientific products. That's one of the, big question marks we had, but we've implemented a bunch of tools into the technology so we can actually get reliable coral mapping data. And so we'll be starting to put out some global maps later this year, and it's a very exciting time. So those are some of the technologies I've been working on. We are looking at, again, testing and developing them on Earth, but ultimately with the eye of exploring some of the other oceans that we know exist in our solar system. We have liquid hydrocarbon seas on Titan, which is that top left moon in the middle there. These are not to scale, so they're not as big as Earth, but these are different icy worlds of Saturn and Jupiter that we know likely have liquid oceans that are larger than Earth's oceans under their ice sheets, or in the case of Titan, under just a methane atmosphere. So it's very exciting because this technology translates not just from Earth, but also to these ocean moons. So with that, I'm going to turn it over to Stephanie, who will take it away.
Thanks, Dr. Sharath. So yes, my name is Stephanie, and I am in the NPS Exploration Science track here at Erasmus. And I am also the Field Campaign Manager with the Aircraft Center for Earth Studies. And today, I mainly want to talk about how an interdisciplinary background and diverse skill sets can really be implemented here in ocean exploration and innovation and in the sciences. So my background is primarily in theater. So I have a Bachelor of Arts degree in theater and philosophy, and I spent 4 years on cruise ships prior to embarking with my career here at Rasmus. And it is through all of those skills that I was able to take on so many roles within the ACES lab. And so the primary role that I have is being the field campaign manager for our upcoming expeditions, and working on our web and logo design, which I have a bit of design from working in a broadcast studio prior to this, handling our scientific visualizations, and then doing something that's really cool and a little out of my realm, which is creating our tidal solar electric research vessel. So with our tidal solar electric research vessel that we are calling TSERV, it started off as an 18-foot kind of pleasure cruise that we took out the lead acid batteries. You can see me there on the upper left-hand side. And the hull of the boat is the smallest person to be able to fit in the space. And so I removed, I think it was 8 lead acid batteries. We put on a tidal generating motor on the back of the boat that is capable of using the tidal flow to charge our lithium batteries. And we put on a custom T-top that holds 3 of our 170-watt solar panels that charge our lithium batteries as well, and then a 50-watt solar panel on the bow of the vessel that charges a supplemental battery. And so with using our TSERV, what we're calling it, it really allows us to transport all of our drones, our fluid cam, all of our innovations that Dr. Shariath has created, and use them here between Biscayne Bay and Key West and mapping our own benthic backyard. And so one of the upcoming expeditions that we have is in Chico Rocks, and we are just very much looking forward to being able to use our own vessel to do the work that we do. And so this is a little video.
Oh.
That doesn't quite work.
Let me try.
But that's okay. So we were able to get and there should be one more slide of us.
Yes.
We were able to get our research vessel out on the water a few days ago and take some amazing pictures and get some great drone video that you see here. Take a look. So that's just a little preview of what we've been working on over the past couple of months. And we are just very enthusiastic about being able to have this research vessel. And I'm enthusiastic about being able to use all of the skills that I have from doing all of the work that I've done over, the course of my short span here on this earth. But just using all of those skills to help bring more attention to the ocean sciences and to get more people involved. So if you have a diverse background or diverse skill set, don't think that you can't, make it work here in the sciences. Thank you.
Thanks so much, Stephanie and Dr. Shariath. That was really nifty, I have to tell you. I'm just blown away with all the different types of studies and research going on, from under the sea to thousands of miles up into space. It's really unbelievable just how far we've come in our fields. It's really neat. I think there were a couple questions in the chat that some of the other folks may have addressed, but please keep an eye out. And I will invite you guys both to stay on if other questions pop up that you might be able to answer for us. So thank you very much.
Thank you all.
Now we're going to move on to Dr. Pakita Zudema, who is going to be speaking to us about low clouds and why we should care about them.
Thank you. I want to thank the organizers and the other presenters for what has been a great morning. I kind of realized that I didn't like my title. They don't really it didn't convey the passion and that I feel for what I work on. So I think this is a little more accurate, like, clouds, the cloudy sky. It's, we all, it's part of the world that we live in. We all experience it. And yeah, we all experience it a little bit differently depending on where we live, and how we go through our daily lives. So here you're seeing kind of a typical spring day in southern Florida. I'm bragging a little bit, but it is kind of wonderful right now. So let's see, I'll let this end and kind of notice what's going on in this video. So this is, I have a camera up here in the roof, and it takes these cloud lapse pictures. And so I use these to kind of give me ideas about what may be influencing the cloud development. So it's it's entertaining, it's beautiful. We can understand it through combining theory, observations, and modeling. This kind of matters because the clouds redistribute the energy that we see coming in from the sunlight. So low clouds can bounce the sunlight back to space, and that helps cool the surface underneath. How that is going to change in a different climate will change, our experience of climate change as in future times. And I was really moved by some of the other presenters who talked about their background and I don't know, issues of diversity in our field. So it's pretty white field, atmospheric sciences. This is, I do field work all over the world. So that's part of what's really exciting about this field. And so this is from a pretty recent campaign that was based in Africa, actually. And we're all pretty white in here, is fair to say. As an older female, I guess, growing up, I didn't see any role models for myself. So I found that really hard about atmospheric sciences. So I'm personally happy to see more women in the field, but we need more people of different ethnic backgrounds too. And I do think that this field actually lends itself really well for integrating people from different backgrounds and places in the world, because our atmosphere is, it's a global enterprise. Like we need to know what's going on in other parts of the world to be able to forecast our weather well here. We can't treat the US like an island. So my own group is diverse, and I like to include people from all over the world for that reason. Also because I think people just care more about the places where they grew up. And a lot of people here are go into atmospheric sciences because they went through a hurricane, and that kind of moved them to want to understand that environment better. But there are, different kinds of weather systems, everywhere, and getting a better understanding of that ultimately will make for a better world. Okay, but the kind of stuff that I do, so the question that we run into with these low clouds, so let me bring up that picture again. You see that these clouds are really thin, but they cover a large area. So when they look like this, they do have a big impact on the energy redistribution within climate. And we have kind of a hard time sort of modeling this well. So that's a big driver for trying to do better. You look at Miami, that actually these clouds kind of mix out pretty readily, and we get a lot of sunny days here. This is another day from the summer, and this looks pretty different. And you'll see why pretty soon. Right, it's raining. The rain shower passes, sun comes out again. But you see how those clouds in the background, they're just becoming way more vertically developed. They're just much deeper. And so the way science works is we pose questions and think of hypothetical answers to them, and then go out and try to answer those and keep revising our thoughts about how the world works until we get to something that seems to work. So there's a big difference between this day in August and the earlier day that I showed you from a spring day. And maybe just try to think of an explanation for what's different between this August day and the February day in Miami that you saw on the earlier slide. So I guess I'm gonna jump ahead a little bit. One difference is that a lot of the water in our atmosphere, we actually cannot see. So this is, another interesting thing about clouds is that they look so substantial, but really it's only like 5% of the water of the atmosphere that's going into the cloud cover. So you just tweak that, percentage by just a little bit, and you can actually get pretty different cloud fractions out there that, in turn interact with the sunlight coming in quite differently. And this video is kind of neat. In the background, you see that, notice how those clouds kind of flatten out at the very top. So there's, that's because it's hitting what's called the tropopause, and that's like a big stability layer on the top of the part of the atmosphere that we live in, and the clouds just have a lot of trouble penetrating that. So how that is going to change in a warmer climate is also something that people are kind of going after. So we sort of have an idea, sort of the basics of clouds development, that a lot of the ocean, a lot of the globe is covered by oceans, 70% of the world. So you get a lot of evaporation that comes off of that. Our breath also puts water vapor into the atmosphere. Trees and vegetation, they also put out H2O. So we have a lot of sources for water vapor. Moist air is lighter than dry air, so it wants to go up. And when it does that, it cools off, and those air parcels will ultimately saturate, and that's relative humidity of 100%. So that's when you get this cloud cover. And this on this day, the clouds are mixing in with the dry air, so that's kind of why they're disappearing. Whereas on this other day, the clouds were able to keep developing. That lets the drops get bigger until eventually they start to rain out. So that was right on time there. So that's what you're seeing there. So we've kind of developed a basic theory about how this kind of process. It's you can kind of encapsulate it as the bubble theory. These clouds sort of look like big bubbles that are growing up. So it's let's see, this is, so as the air moves up, the drops keep accumulating vapor, so the droplets keep growing. Something that cloud physicists have struggled with for a long time to explain is how you get from pretty tiny drops to raindrops. There's a schematic here in the lower left-hand corner here. It looks like a typical droplet is about 10 micron. A human hair is about 100 microns. So a typical cloud droplet is about 1/10 the size of that, whereas a raindrop is about 1,000 microns. So it has to grow by 2 orders of magnitude And it does that in a pretty short time. I mean, you're seeing that in these videos that you can start with a, you know, something that doesn't look like much. And then really within the space of about 10 minutes, you can get a pretty good soaking from one of these clouds. So so the way people explain this now, but this is still actually something that people are working on, is as these clouds drop, cloud drops grow, you get something that's called, you know, there'll be one droplet that becomes big enough that it starts to fall. The lucky drop, you can call it. And as it starts to fall, it starts to collect all these other smaller drops on the way down. So you end up, once it starts falling, like you see it in the schematic in the lower right here, it can actually accumulate a lot of the smaller drops pretty quickly. And in that sense, you get to the raindrop pretty fast. So, but this is a very singular way of looking at a cloud. Clouds do tend to come in groups. And I think towards the end of this clip, you'll kind of see something that we call, no, not this one. I mean, here's a picture of it from space, but when you do have clouds out there, they like to organize. You know, they're rarely by themselves. So once they start to organize, then maybe that part sort of helps clouds grow and develop a little faster. So, and that's actually something that that I research with. So this is Shujin Li, she's from China. So once you start to get the rain and you get these gust fronts, you may have felt them sometime yourself when you're out and it starts to rain and you just feel like this wash of cold air that comes with it. Like, well, that cold air actually helps organize where the clouds are spatially around it. So, and that can actually help develop the clouds further. But other questions that my group has looked at is interactions with smoke. So this is over, you know, smoke is starting, wildfires, that's starting to become a big topic. Like we are seeing more and more fires every year, and those can change the clouds that they interact with. At Miami, we have dust that travels across the Atlantic and reaches us in Miami. So that, you know, which is several thousands of kilometers. So that's pretty intriguing. How does it get here? What is it like when it gets here? Yemi he also looked at how smoke interacts with clouds and how that depends on meteorology. Isabel looked at how clouds respond to changes in the ocean sea surface temperature, because we expect that to change. Mampi Sarkar in the lower left here looked at how clouds evolve between California, where you get the stratus clouds that are very you know, overcast but thin, but then they turn into clouds more like what we see here in Miami. So this is kind of the research stuff, but there's really a lot of other exciting, meaningful jobs in atmospheric sciences besides academia. And I really think this is a growth field because there's a lot more interest in microclimates. So, you know, more sports teams are having their own meteorologists, airports have their own meteorologists, lightning, people really want to predict when and where lightning occurs, which used to seem fantastical, but now it's starting to be doable. Something called forensic meteorology, lawyers hire meteorologists to, I don't know if there's a lawsuit about a crop failure or something like that. Energy use forecasting is already large, and it's going and growing fast, like how to predict wind and solar energy use and climate change impacts at the neighborhood level. Certainly in Miami, that is, there's a big effort by the Miami community to understand how the urban heat island interacts with you know, changes with sea level rise. So with that, I'd like to thank you for your time, and yeah, feel free to put questions in the chat and reach out to me.
All right, thank you very much, Dr. Zedima. Folks, if you have questions about clouds or about atmospheric sciences or some of those potential career fields, please, I believe Dr. Zedima will be staying on with us for at least several more minutes so she can provide further information about some of those. If I may, I'm curious to know, what have some of your former students, what have they gone on to pursue after they have worked with you? Types of career fields, that is.
Yeah, so they have tended to stay in research and academia. I have to admit, because that's kind of what I train people to do. Yemi is now an assistant professor out in California. Two of my students, actually, they work a lot in the labs. So Mumpy Sarkar is working out at NCAR, National Center for Atmospheric Research. So she's also continuing to work on aircraft campaigns. Yinghao Shang, so he is doing research on marine geoengineering. So this is another topic. So people are looking at nucleating clouds to make them brighter, so as a way to combat climate change. So he is researching where the best places are in the world to do that. Let's see, Xijing Li is working for NASA Langley on looking at Arctic clouds. So that's in Hampton, Virginia. And Samantha Kramer is working for an air quality company out in California.
Wow, you must be pretty proud to have that, your network of your former students throughout the country contributing in this field.
I am. I think it's the most satisfying aspect of my career.
Oh, well, I have to say, I couldn't have asked you for a more appropriate closing quote. So we're gonna start to transition now. I want to thank you again on behalf of all of us, the presenters, the moderators here at Erasmus, as well as the participants online. Thank you, Dr. Zedima, very much.
You're welcome.
I'm going to officially take off my moderator hat now, everyone. It's been a pleasure working with you, and I'm going to pass the baton to the moderator who will be assisting you for the rest of the afternoon, Aaliyah True.
Good afternoon, everyone. My name is Aaliyah True, and I am going to be your host for today, for the rest of today. And thank you again for all the previous presenters who gave riveting talks. That was really interesting. Just as a quick aside, I'd like to introduce myself and my background. I am a PhD student in Dr. Nikki Traylor-Knowles' Cnidarian Immunity Lab. And it is actually my first year here at Erasmus, and it has been a wonderful year, a bunch of new experiences. I originally come from Texas. I did my undergrad at Texas A&M University, and I was a fisheries biologist there. So a bit of a change to do coral immunity. And for those of you who have stayed since the talk, the second talk about diversity, where Natalie spoke of taking up space, I would like to also say that that's something I really resonate with. I come from a background of poverty. I come from a background of being in the BIPOC community, Black and Indigenous. And it is a hard battle, but keep fighting, keep up the good work. And with that, I'd like to introduce our next speaker, Kelly Saluri and Kristen Best-Otubu on fisheries research.
Hi, Aaliyah. Thank you so much for the introduction. And I'm going to share my screen now. All righty. Can everyone see my screen? Perfect. So thank you so much from learning from everybody's research. It's been really interesting. And today I'll be talking about quantitative fisheries research in the Babcock Lab here at University of Miami. My name is Kelly Soluri. I'm a first year master's student in Dr. Babcock's lab. And I'm also joined by my co-lab mate, Kristen Besto-Tubu. If you'd like to introduce yourself, Kristen, go ahead.
Hello, good afternoon, everyone. As Kelly said, I'm also a second-year PhD candidate in Dr. Beth Babcock's lab, and I'll be discussing my path in fisheries science from a fisherman's point of view. So I'm excited to share that with you guys.
Perfect. So let's get started. So my background, growing up, I didn't have a lot of these field work or lab work skills that so many interesting labs are able to do until I got to college. But I graduated in the year 2020. And so when I graduated college, I didn't really have a lot of opportunity to go into jobs with field work or lab work skills. And so I plunged myself into analytical skills, which I ended up really loving. And so analytical skills are really important. We live in an information age where there's so much information out there and not a lot of people that can break down that information to understand what it actually means. So the photo below can just look like a bunch of numbers to you, but if you have analytical skills, you can really pry apart these numbers, find the story, and actually get down to any conclusions that you might have from these numbers. So I'm gonna go over a couple steps to analytical skills that we use here in the Babcock lab. Exploratory data analysis, modeling, like machine learning, and data visualization. So exploratory data analysis is kind of like having a first impression when you meet someone. You ask them questions and it kind of makes you understand their characteristics, what they're like. And so that's kind of what you do with a data set. You first ask a couple questions, maybe some summary statistics, get to know like what's your smallest value, your largest value, your median mode. You ask maybe the range of your values and maybe have a couple of initial graphs just to really kind of understand what you're looking at, the characteristics, and maybe some kind of data trends that you may notice that can affect the path of what kind of questions you're going to ask with your data. Modeling is a process of using mathematical equation to explain a phenomenon. And modeling is really cool because a lot of the times you can model things that you can't observe in nature, and you can really find that through numbers and through math and trying to see different variables and how they affect the phenomenon. And so here are two photos of modeling that I've done. And you can see how the line of best fit might show like the phenomenon better. Machine learning is a hot topic. It's a type of artificial intelligence or AI that allows systems to make decisions, identify patterns, and learn from data. And if you've used social media or used your search bar, you have been in contact with machine learning. If you look for something online, the algorithm will say, this person is interested in this, so I'm going to recommend things that are similar so they can click again. And so that's kind of like what's showing in these photos here. On the top, there's a correlation matrix trying to see the different aspects of different cereal nutritional content. And I was trying to analyze what are the similarities between brands. But instead of going into the very difficult analysis of looking at each nutritional fact of the cereal, I trained a machine to be able to cluster them together already. And so the machine is kind of doing that work for me. And you'll see that below in the cluster model. And data visualization, I think, is one of the most important parts of analytical skill building, because what's the point of having a really strong analysis if you can't communicate that to others and you can't share your conclusions so they can make the best decision for them. And so you can use things like this presentation, different graphs, software like Tableau to help you really visualize your results best. Here is shown like a dashboard that I made in ArcGIS showing different fish surveys in Biscayne Bay. And so these skills are applicable to any field that uses information to make decisions. And that's a lot of different fields, but you're here because you're interested in marine science. And so our lab uses these tools to understand fish populations and their biology and their populations. And so here's a couple of photos. To the left is an LMR CSE undergraduate project presentation, and we'll talk about that program a little further down. And Adrienne, one of our PhD students, doing lab work and us doing, or she's doing field work and we're doing lab work together. So I'm going to go into a couple of the research topics that we cover here in the lab so you can see the diversity of the different projects we have using these machine learning and data analysis tools. So Adrienne is a fifth-year PhD student in our lab, and she researches lane snapper populations. She's trying to see how old the populations are, how they grow, and how they're connected genetically in the Gulf of Mexico. And she's using all these different quantitative models to help manage these populations better. And she is a Living Marine Resource Cooperative Science Fellow, or LMRCSC Fellow. And here you see an otolith on the top, which is a bone structure in fish ears that help you understand how old they are. She's also doing field work, and her photos show her in Belize, which is her study site. Marissa is a fourth-year PhD candidate, and her focus is on bycatch, which is when you go out fishing, the fish that you don't mean to catch, and that's not your target, that's called bycatch. And sometimes sea turtles can be bycatch. And so she's trying to see if bycatch methods accurately help the management of our sea turtle populations, specifically loggerhead sea turtles. David is a second-year master's candidate who is defending soon. And he's kind of looking at the methodology of science and seeing how little can you sample in order to get the most information. And he's using that for mangrove fish surveys. Ashley is a second-year PhD candidate in our lab, and she has a really cool project where she's using a type of artificial intelligence or machine learning called deep learning to create a call detector for Rice's whales in the Gulf of Mexico. And so she's trying to make this machine so it can pick up the calls of these rice whales and try to understand how they move over time. And so, oops, sorry about that. And so I'd like to introduce my lab mate, Kristen Besto-Tubu, who is a second-year PhD candidate, to talk about his experience, how he got into fisheries, and more about his research. Take it away, Kristen.
Thank you, Kelly. So first of all, my path in marine science and specifically fisheries science all started from when I was very young. And when I walked around the lakes and ponds with my parents, I noticed all these guys with fishing rods, having catfish and bluegill in the bucket. So I've been a fisherman since I could walk. It led to a need to understand the fish, to understand what do they eat, where do they live, what's the best time of the year to catch them. And so most importantly, besides going to the library, buying books, and watching fishing shows in the morning, I talked with these local fishermen that have been fishing at these water bodies since they were my age at the time. And the longer I fished at these water bodies, the more changes I noticed, such as algae covering up the ponds, the rivers becoming muddier, and bait becoming harder to find. Where before I could catch some minnows in 10 minutes, 20 minutes, now it took an hour or two and I had to walk further around the lake. And in order to try and deal with the worsening of some of my fishing spots, I also had to go to more secluded areas where there weren't as many people and there wasn't as much pollution in the water bodies. And so this led to a lot of questions when I was younger, like, why is this happening? And so my true research really began when I went multiple summer internships in my undergrad. These are just the three that I went through that I highly recommend. First is the LMRCSC sponsored University of Maryland Eastern Shore RU. Then there is the Blandy RU at the University of Virginia, and then Woods Hole Partnership Education Program. And it's through these internships where I began to understand the processes that influence fish populations, aquatic insect populations, and it also helped me identify ecology as my preferential field of study, where I look at the relationships between fish and their environment and what do they eat, rather than biology or chemistry. So these were just a couple of pictures taken at my internship sites. On the left is me showing the local summer camp some of the aquatic insects I was collecting, and I mentioned how these dragonfly larvae are important to supporting trout populations. And on the right-hand side was when I worked at the Northeast Fisheries Science Center in Woods Hole PEP, looking at how diet and environment affect summer flounder condition factor, which is basically how full or how large a female summer flounder is to have eggs. So my current research at Erasmus, I'm looking at the Biscayne Bay, which is underwent major changes in water flow and habitat due to canal dredging and increasing urban population. And I want to see these functional group responses, functional groups comprising of important fish populations, whether they be recreational fish or just ecologically important populations, by using multiple food web models, in particular using the EcoPath ecosystem modeling framework to look at the temporal, meaning over time, and spatial aspects of these population dynamics in the bay. And this is just an example of an EcoPath model, which is like a picture of the energy flow, where each level on the left-hand side, you'll notice on the y-axis, numbers 1 through 5, that represents the trophic level. The bubbles represent the functional groups, and the lines connecting them are the trophic linkages. So how do the energy flows? So I'll be using models such as these in my research. And for future direction, I would like to bring more people into the marine science fold. In particular, I'm thinking about starting a University of Miami fishing club, just to introduce people into the environment the way how I got introduced from a young age, because it's important to be a part of the ecosystem yourself in order to gain a love for it and for you to get an understanding of it. So thank you for my time.
Thank you so much, Chris. And it's really cool to hear about your story. And so here you can see so much of quantitative and analytical research used to learn about a lot of ocean processes and quantitative fisheries. And it's really interesting for me. I think it's kind of amazing that you can do it from your computer. And so you can do it from really anywhere, even if you don't even have ocean access. You can still have a marine science career. So I'd like to show some free resources for analysis skill building. So you can learn coding and analysis skills for free. Data Science for All program is an 8-month boot camp for people of color, LGBTQ+ folks and veterans. And there's another program for women in data science. It is completely free. I'm an alumnus of this program. That's how I learned Python and SQL and different computer languages, and I highly recommend it. And if you are in the Miami-Dade area, and maybe you do dual enrollment in your high school, or maybe you go to the community college, this program gives you credits towards a data science degree at Miami-Dade Community College. And there's also free or affordable certifications from Coursera, LinkedIn, or Google, and you can scan to learn more about these opportunities. And there's also a lot of resources for marine science for people who are minorities. And so we've talked a lot of people in our lab are part of this LMRCSC program, which is a Living Marine Resources Cooperative Science Center. They give you tuition for your graduate degree if you're thinking of going to grad school, stipends. They give you networking opportunities to talk about people who are further along in their career, a conference for you to show off your research, and training for careers in research management and public policy. So outside of academia as well. We also have Minorities in Shark Science, also known as MISS, that gives you hands-on field experiences in shark research, different fellowships, personal development, and community building. They also have scholarships and funding. And if you're in high school, they also have programs for high school students as well. And then some of the different diversity programs in NOAA and their educational partnership program. And so you can scan any one of these QR codes to learn more about their respective opportunities. And so thank you all to all the current members of our lab, the alumnus, and our friends for the photos and descriptions of their research. We hope this has inspired you in your career for marine science, and hopefully you can use any of the resources to help you further along in your career. And if you have any questions to any one of the researchers shown today, here's everybody's email. And so feel free to reach out and ask questions. Thank you.
Thank y'all so much for that presentation. I know when I first started in fisheries science, I had no idea how much analytical data I would have to be going through. So it took a lot of catching up. We have a question in the chat from Adriana. At the fisheries, what temperature and level of water do you put the fish in?
Would you like to answer that question, Kristen, or should I?
Sure, I can take a crack at it. So for my research, I'm not doing a lot of lab testing where I put fish in temperature or level of water. Instead, I'm seeing how the salinity and nutrient concentrations of the water will impact fish populations by modeling, say, how the populations have decreased due to increasing salinity or due to increasing nutrients. So mine is looking more at it from a model point of view.
Awesome. Thank you so much. And again, I'd just like to reiterate that these two will be staying on for a couple more minutes for any extra questions in the Q&A. Thank you. Also, there was one question I'd like to bring to light, and I'd like to say it out loud just in case. Catherine asked if there are any mentors in any of the programs on campus, and one I would like to say that we have is the Empower Me First mentoring program. And this is for undergraduate first generation students that can meet with faculty or staff to assist in building social capital from on campus and beyond. And I know that in my career, I would have loved to have had this opportunity or at least known about it because it greatly helps build your social capital and just create a community. So up next, I'd like to transition to Dr. Nikki Traylor Knowles. She is going to speak about Black women in ecology, evolution, and marine science. So take it away, Nikki.
Okay, hopefully you all can see my screen and everything's good. It's wonderful to be here and to talk with you a bit about the nonprofit that I started recently called Black Women in Ecology, Evolution, and Marine Science. So a little bit about who I am. So first and foremost, I'm a wife, a twin mother. I am a mother of 4-year-old twins. I'm also a daughter, and I'm a sister. My family is the most important thing to me and something that really helps to guide the work that I do. I'm also a scientist, a mentor, and a teacher. So I'm an assistant professor at the University of Miami, been here since 2016. And my laboratory works on coral immunity, which I'll talk about very briefly today. And I'll give you some resources to reach out if you're interested in learning more. I'm also an advocate, and I'll talk about this a lot today and about the work that we're doing to help change science and make it a more equitable place for not only Black women, but for everybody. And then also recently I've become a special collections director. We have the Voss Marine Invertebrate Collection here at University of Miami Erasmus. Unfortunately, I don't have time to talk about that today, but hopefully in the future, I'll be able to talk about that more. So here's my laboratory information if you're interested. We're on Twitter. You can also contact me directly or check out our website and connect with some of the cool stuff that we're doing. So what has always gotten me interested in marine science is that I've known since I was 9 years old that I wanted to be a marine biologist. And it wasn't until I was in college that I discovered coral reefs. And the reason that I got so into coral reefs was just because when you go diving or snorkeling and you look on a reef, every time you go there, you see something new. There's always something new to discover. And then once I started learning more about it and learning how important they are, it just became this passion. So my lab is really interested in the coral immune system. And in particular, how does the immune system of corals help it survive? So I'm sure many of you are familiar with coral bleaching and coral disease. And particularly here in Florida, we're dealing with a lot of these impacts. So my laboratory is really trying to understand, how can corals survive this? Because some corals can, and we don't know why. So we do a lot of almost like medicine for corals in my laboratory, where we apply different types of techniques that we use actually in human medicine to corals. So if you're interested, like I said, please reach out. I'd love to talk. I could talk for hours about corals. And if you're also interested in how to get into science, I'm always here to chat about that. So I want to give a backstory of my nonprofit and just start from the beginning. So I'm an academic through and through. I've always known that I wanted to do this job, but I was getting really tired. I was always hearing that I was tired of being told that Black women don't exist in marine science. I was tired of programs being created, which I didn't think actually addressed the problem that we have. And I was also tired of being the only one in the room. So many times throughout my career, I've been the only Black woman in the room, or even the only person of color in the room. And that gets really isolating. So like any good scientist, what I did was I reached out on Twitter and I just said, you know, I'm a Black woman marine biologist. Are there any others out there? Like, let's connect. And And from that, it started off this huge wave of connection. And so we started this list where, we started listing people of who, of women, Black women that were in marine science. And so from there, we got a list of a couple hundred scientists. And so we had our first virtual meeting in July 2020. And so it was really wonderful. We had all these women on the call. And for once, I felt like, oh my God, I'm not alone. There's so many of us. And so from there, it was like, you know what? We need to develop a program around this to help facilitate community. And based on having these virtual meetups, what I discovered is that there's a lot of needs. And in particular, and I'll talk about what demographic we're actually targeting. But what I saw was that there's needs for community, there's needs for resource sharing and training, for financial support and networking, mentoring and sponsorship particularly. So this is the behind the scenes advocacy that's so important for progression in your career. Also for creating a new system for success and changing the narrative that we are here and we're doing amazing science and we deserve to be recognized. So our target demographic within BWEAMS, that's our nickname, is Black women from the undergraduate age all the way through to senior scientists and emeritus, retired scientists. All events and resources are free for BWEAMS members, and it's free to become a member. Our age ranges between 18 through 54. And the biggest reason why this space is needed is because Black women are isolated in science. And they have to navigate a system that's been historically designed to exclude them. And so we're really here to change that and create a community so that Black women can feel like they belong, because they do. So our mission is to provide a network for creativity that embraces innovation and equitable collaboration for Black women. And our vision is to provide a crucial platform for Black women scientists to inspire and lead. So what we actually do, like on the court, what are we actually doing? So we do a lot of monthly events, from panels, workshops, science shares, where we have a safe space for Black women to share their science with each other, coffee hours where we can meet with different folks in different fields that are, at the head of their fields, and to meet and network with them. Community meetups, which are just community meetings where we have a safe space for Black women to vent and support each other. And then happy hour and special events. And other programs that we also have, last year, we piloted a mentoring program that was really successful. So we're working on expanding that. We're also going to be working on doing more outreach. So starting to work with other programs that are doing programs with kids and folks that are beyond the age range that we're in. We have a lot of opportunities for membership networking. We also have a Slack group for the BWEAMS members where they can network and get information very quickly. And then we also have a weekly writing group. So this is really important, in particular for those of us that are in academics, where writing is basically the currency by which we're judged. And so we have this weekly writing group where people can get together, work together, and work on achieving their goals. So in 2020 alone, we reached over 450 Black women scientists through about 50 events. So we're really excited about this and really impressed with what we were able to do so quickly and with such a small budget at the time. And it really does take a village in which to do this. So we have a board of 7 members. And then we've recently have been expanding our staff. And so I just want to give a shout out to all of them, because without their help, none of this would be happening. I'm really great at ideas, but I can't do everything. So their help and their ability to actually see things through is why we've been a success and why we keep going. And so where we're going in the short term, we're trying to expand. We want to start hiring more staff. We want to create a grant and fellowship program, in particular for undergrad to graduate student level. We want to expand our mentorship program to provide more support for women undergrad through career professional levels. Also developing a coaching program, develop internship opportunities, start to have in-person local chapters and meetups. And we're in the process of planning a in-person conference, which we're really excited about. And soon I'll be able to announce more about that. And then long term, our goals really are at the heart of really dismantling the white supremacy culture that often is a big part of science. We want to create a new system where everyone can flourish. So we want to make it more equitable and a place where everyone feels safe to be able to do their work. So with that, if you're interested, if you or someone you know identify as a Black woman in ecology, evolution, and marine science, please reach out. Membership is free. All the events are free. And it's a really amazing space for you to find people and to connect. So we're on Twitter and Instagram. This is our email. You can also connect and sign up through our website. And lastly, if you'd like to donate, please, there's our QR code right here. And with that, I thank you so much for your time and am happy to take any questions. And I'd like to thank all of our funders. Thank you so much.
Thank you, Nikki, for that wonderful presentation. We have a question in the chat. What were some of the biggest struggles as a Black woman in marine science, and how did you get through them?
Yeah, so I mean, we could talk about this for a while, but to really keep it short, I think that, you know, it's hard when you're in a system that really wasn't designed to help you. So oftentimes I would feel very isolated or feel like I was like I didn't belong or like my ideas weren't valued. And so what I did to move through those was, one, I had to create my own network of people that I knew I could trust. I had to learn to develop my voice, which took a while, but it did develop. And I think I also really relied on you know, external support. So from my family, mental health support, also my friends, you know, outside of science, to know that I'm not crazy and what I want to do matters. So yeah, but Jasmine, if you also want to contact me, I'd also love to talk with you more one-on-one if you're interested.
Thank you again.
Yeah, thank you so much.
So up next, we have Dr. Danielle McDonald, and she's going to be presenting on serotonin, totally dynamic. Welcome.
Thanks, Aaliyah. Can you guys hear me?
Yep, you are good.
Thank you so much. Hi, everybody. My name is Dr. Danielle McDonald, and this is the Rasmus Toadfish Lab. We do a lot of different things in this lab, but today we're going to talk to you about our work on serotonin. Serotonin is a chemical that's found in the brain and throughout the body of humans. And in fish, it's involved in regulating heart function, gill function, and the response to environmental hypoxia, which is low environmental oxygen levels. Now, hypoxia can occur in the environment naturally, but it's also occurring a lot more often due to pollution and climate change. Come, let me show you my lab.
Hi everyone, my name is Mary and I am a third-year master's student here in the lab, and it super excites me to say that I will be
Oh, we are missing your sound.
We got it. Okay. Hello again. My name is Mary. I'm a third-year master's student here in the Toadfish Lab, and it excites me to say that I will actually be graduating this spring, which is one of my biggest dreams and hugest accomplishments to date. So I'm originally from Los Angeles, California. I was raised by a single mom, and it was just me, her, and my brother growing up. We didn't have a lot of money, grew up in a rough neighborhood, and so I had to deal with a lot of trials and tribulations of that. But I just want to tell anyone who is here today that no matter where you come from, what your background is, that you can achieve your dreams. And if that is to pursue a career in science, that through hard work and dedication, that you'll be able to succeed. And as my mom tells me every single day, si se puede. So let's go ahead and take a look at a toadfish. So this is a toadfish. It's indigenous here in Florida and Biscayne Bay, but is also found all throughout the Gulf of Mexico. And we keep them in these aquaria. And so you may be looking, well, why is there PVC pipe in there? And so in their natural environment, toadfish tend to be bottom dwellers. And when they find their home, they'll actually make it inside of kind of these rock burrowing formations. So we here in the lab try and simulate that to the best of our ability. So when we work with toadfish in the lab, we will typically put them under anesthesia. And so we can take a quick look at a toadfish here, but I'm gonna go ahead and pop them in the anesthesia. And so we do this not only so that we're able to safely perform procedures, but that also it's safe for the animal as well. So this takes a couple of minutes usually. So I already have a toadfish that has been in anesthetic. So if we take a quick look, we can see that they are scaleless. And this is what we believe that allows them to be a very hypoxia tolerant animal, meaning they can survive in low oxygen states. And if we take a quick look in its mouth, we can see that it has these kind of very sharp, tiny teeth. And so they are very vicious and ferocious, we like to say in the lab. So that is also another reason why we use anesthetic when doing procedures. So one of the procedures that we actually do is we sex the animals. So let's go ahead and talk to Anastasia, who's going to explain a little bit more about how and why we do that.
Hi everyone, my name is Anastasia and I am a second-year PhD student here in the Toadfish Lab. So I was originally born in Ukraine and I immigrated to the United States with my family when I was 5. One of the things that I really enjoyed learning about growing up was the ocean, but also how things work in the human body. So it's no surprise that I ended up here because we do physiology research. However, I never pictured myself as a scientist per se, because I always thought that I was better at writing and communicating and disciplines like that. But something that I want to say to aspiring scientists is that creativity is a big part of the scientific process. And research and writing and teaching people is all something that scientists should be very good at. So one of the things, like Mary mentioned, that we do is ultrasound the toadfish. So I have a toadfish here in the water, and to ultrasound the toadfish, we use this ultrasound machine that you can see in many different hospitals that are used for ultrasounding babies in the womb. So what we do is take the toadfish, flip it on its back, and put the probe up to the abdomen and try to see if there are any eggs, and that will determine if this is a female or not. So I don't see any eggs in this one. So I'm going to say that this is a male. And the reason that we do this is because there is no other way to identify whether the fish is a male or female other than the ultrasounding. So around early January, the female toadfish will actually get gravid. And their abdomen will bloat and they will have these eggs, which are indicated by the black dots here. And so once they are gravid, what we will do is place them with a male into our breeding tanks. So we have our breeding tanks over here with the adults, and they're actually sectioned off by a black just garbage bag, so they can have some privacy and peace and quiet. So the female feels free to lay the eggs, and the male can go ahead and fertilize them and actually stay there and guard the eggs in the shelter until the fish are fully hatched and free swimming. So one of the things that we do with the fully hatched fish is other research that Shannon will tell you about more.
Hi everybody, my name is Shannon and I am an undergraduate here at the University of Miami studying marine science and biology. And so one of my favorite things to do is to be outside in nature. I've always loved the outdoors, and that's what really inspired me to get involved in science. And so what I would specifically love to do is to help animals and to help the environment. And so I hope to be able to combine my love for scientific research with conservation efforts to be able to create a positive difference through science. And so you guys just got to see some adult toadfish, and now we're going to take a look at some baby toadfish. So let me catch one of them for us today. These guys are very fast, as you guys can see. So we're just going to try to capture one. Whoop, we got a couple actually. So you guys can probably notice that these babies look very similar to the adults. We got 3 in here. You can kind of count them out there. These are 3 fish. Again, these guys are both toadfish, and our scientific name is Apsanus beta. So feel free to try that out, Apsanus beta. And these guys are marine teleost fish. That means that they are bony fish that live in saltwater conditions. And so these guys are great model organisms, and we use them in this lab because they're very abundant, and they are great at being hardy fish, so we can look at a lot of different environmental stressors with them. And so one thing we're really focused about in this lab is understanding something known as the urea pulsing mechanism, and that is controlled by serotonin. And it's exactly what it sounds like. It's when fish expel urea into the water. And so what my project is focusing on is understanding at what age do these fish pulsate urea. And this is really important so we can understand whether this chemical communication between fish is related to more social status or more reproductive status. So that's some things we're hoping to learn more about.
Okay, and now we're going to talk to Oriana about her research.
Hi, like Danielle said, my name is Oriana. I'm a 3rd year master's student. I have been in love with science my entire life. Growing up, I was lucky enough to have good role models in science and in education. My dad was an environmental scientist my entire life, and both him and my mother have master's degree in their respective fields. When my dad was out doing work in the field, in the rivers or in marshes, I would always be there with him, you know, playing with the nets, acting like I was really catching things for his research. I would go home with him and we'd look under the microscope at the water samples that we would pull up. They're always very murky, very dirty because we're pulling them out of dirty streams. But it was always really cool to see the little organisms swimming around, all the little bugs and worms and tiny little fish. And so seeing that growing up brought me into my love for marine biology and marine science and studying the physiology of the marine teleost fish, the Gulf toadfish. And so my research is focused mostly on the neuroepithelial cells in the Gulf toadfish. And what those are oxygen sensing cells. And they are very helpful in the hypoxia response of the toadfish. And so like Danielle mentioned at the beginning, hypoxia means low environmental oxygen. And so the neuroepithelial cells will sense the oxygen is dropping in the water, and it'll cause a response in the fish depending on each fish on how it responds. And so the way that we look at neuroepithelial cells is by looking for the serotonin in the cells themselves. And so here you'll see a picture of the gill, and these bright green dots all along here are our neuroepithelial cells. The reason they're bright green is because they are fluorescing heavily with serotonin and making us able to see them under the microscopes. What we're seeing in this image is the neuroepithelial cells all along the filament of the gill, which is the main structure of it that holds all the lamellae, which are these green branches, in place. So the lamellae pick up the oxygen from the water, bring it into the gill, and then out through the fish. And you'll see also that the filament itself is kind of bright orangish, and that's because there are nerve fibers within the filaments that are picking up signals from the lamellae, from the neuroepithelial cells, from other cells in the body of the gill, and sending it throughout the fish so that signals can be sent. So one of the other things that we do when we're looking at the neuroepithelial cells is determine their size and exactly where they are in the filament. So we can do that with something called a confocal microscope, which allows us to see a 3D image of the gill. So here you'll see in this GIF, the neuroepithelial cells are coming in and fading out and coming in and fading out. And so what that's showing you is that the neuroepithelial cells are either closer to the top of the gill or at the bottom of the gill. And that tells you that they're different, they're distributed a little differently, but they're all still within this main filament. and decently about the same size. And so what neuroepithelial cells will do when they sense the low oxygen is change the heart rate as well as the breathing rate of the fish so that they can survive in the hypoxic environment. And so that will lead us to John's research with the cardiovascular response.
Thanks, Oriana.
Hello, everyone. I'm John. I'm a fourth-year PhD student here in the Toadfish Lab. A little bit about myself. I'm originally from Buffalo, New York. I got my undergraduate degree in biology, and then I wasn't sure if I wanted to have a career in science or not. So I took some time off, looked around, and eventually I decided to come back to it to get my master's, and now I'm going to be graduating with my PhD this June. So the best advice I can give to a young scientist is that if you think you're interested in pursuing science, to get involved and to start looking for opportunities. But if you're not sure, it's okay to take some time off too. Don't be afraid to move at your own pace. It's never too late to start, and it's never too early to start looking either. So make sure that you keep that in mind. Now, in regards to my research, you've heard a little bit about hypoxia and you've heard a little bit about serotonin, and it's my job to figure out the relationship between the two. My research mainly focuses on serotonin circulating in the bloodstream. So all vertebrates, whether they're toadfish or humans, we have serotonin in our blood. And we think that this circulating serotonin is one of the major players in the hypoxia response that helps keep organisms safe and alive during periods of low environmental oxygen. And in order to test that, we've got an experiment set up over here that I'd like to show you. So what you're looking at on this computer screen is a live look at a toadfish's heartbeat in real time. Each one of these peaks that you see is a contraction of the fish's heart. And so we're able to measure that with this computer program that's hooked up to a toadfish. And we can see how fast the heart is beating, and we can see the blood pressure of the heart and how that changes in real time. The red one is looking at blood pressure in the heart itself. The blue one is looking at blood pressure in the tail. So one of the things that we're interested in looking at is, are these changes that the fish is experiencing during hypoxia universal throughout the whole fish, or are different parts of the fish responding differently to hypoxia? And then down here in the bottom, the green line, this is measuring breathing rate and depth of breathing, which is something that we expect to change during hypoxia as well. So normally when toadfish are exposed to hypoxia, we see their heart rate decreases, their blood pressure decreases, their breathing rate decreases. And what we're interested in looking at is seeing if serotonin is involved in this. So to do that, we're taking another group of toadfish and we're going to be treating them with Prozac, which is a common prescription drug that you might have heard of used to treat depression. Prozac works by preventing uptake of serotonin through serotonin transporters. And so if we inhibit that by treating a fish with Prozac, we believe that that's going to impair the fish's ability to deal with hypoxia because we've taken away their ability to regulate serotonin. And so we're going to be looking at fish that have been treated with Prozac, comparing them to normal fish and seeing how their hypoxia responses differ to determine if serotonin is involved in this process. And so you can take a look. This is the fish that we're looking at. He's a live fish just sitting in the box. He's just chilling, having a good time, and we're able to monitor changes to his heart rate in real time as it's happening, which is a really cool experimental setup that we have. So we wanted to say thank you all so much for joining us today. If you have any questions, make sure to leave them in the chat. We hope you had fun. We hope that you're interested in what we had to say. And thanks for joining us.
And have a totally awesome day.
Thank y'all so much for that presentation. It was so cool to see y'all's lab in action. So we have a question in the chat specifically for Sharon. What kind of field specifically do you plan on going into or are already in with your interests?
So something I'm really interested in getting more involved in would be something related to ecology. I learned my favorite aspects of marine biology are marine ecology. And so right now I'm focusing on, I'm a senior right now and looking into grad programs that specialize in marine ecology.
Thank you. And actually, I have a question, a general question for any of y'all. Do y'all have any warnings or advice for recycling meds to keep them from the waterways?
So yeah, this is a problem, pharmaceutical pollution. A lot of people, when they're done with their medications and they may take their treatment course and have some left over, they'll flush them down the toilet, they'll throw them in the garbage and get rid of them. And that ends up in the wastewater, and that can go out into sea, and we can actually detect Prozac in some locations in detectable levels out in the wild. So if you have extra leftover drugs and you're not sure what to do with them, I believe some local police and fire departments have some prescription drug pickup locations, and pharmacies do as well. So if you have some prescription drugs left over,
Don't flush them.
yeah, just don't flush them down the toilet. That's the big problem. If you're not sure, contact your pharmacy.
Thank you again. That was great. Also, they will be staying behind to answer any following questions in the chat. All right. So now we're going to transition to speaking about chromatography and marine science. And this presentation is going to be presented by Paul Voital. Welcome, Paul. Paul, I believe you're muted.
Hi, everyone. My name is Paul Voital. I'm a third-year PhD student in the Department of Ocean Sciences here at Erasmus. And today I'm going to be talking about chromatography in marine science. I want to start off by talking about what chromatography actually is. So if we look at the word chromatography, we can see that it is made up of two parts. The first is chroma, which comes from Greek, and in Greek it means color. And then the second part is graphy, which comes from the Greek graphene, which means to write. And we now understand that it is used as a suffix to denote the study of something. So literally it translates to color, to write color or to study color. And it was originally invented by an Italian scientist who was trying to separate pigments in plants. So the things that make plants different colors. So it has its origins in separating different colors. Nowadays, we use chromatography to refer to the science of separation of complex chemical mixtures into their different components. There are many different types of chromatography, but all chromatography has three components. The first is a stationary phase, which in this case, in this drawing, is the paper, which is the tannish rectangle. The analyte, which is what you're trying to separate. This would be our sample, and in this case is the colored dots. And the mobile phase, which is the liquid that the paper is sitting in. And the interaction between the mobile phase, the analyte, and the stationary phase is how we get our separation. So in chromatography, mixtures are separated based on chemical properties. So they can be separated based on size, polarity, solubility, volatility, and chirality, to name a few. You can actually try chromatography at home. What you're going to need for this experiment is a marker or a couple markers, depending on how many colors you want for this experiment. A pencil, a straight edge like a ruler, or a cover book, a piece of paper, a cup, and some water. And what I've done, and what you want to do, is cut out from your piece of paper a segment of your paper that fits in your cup. And then at the bottom of the paper, you're going to draw a line in pencil. On that line, you can draw circles or squares. I've chosen squares of color, of each individual color, and you want to space them out so that they don't run into each other. And you want to fill in those shapes, whatever shape you have chosen,
so that you have maximum. So I have a slightly different setup than you will at home. You'll just need a cup at home, but this setup is just a little bit easier to demonstrate how this will go. So I've taken my pieces of paper and taped them to a vial. You can just place your paper in the cup and the cup will keep it upright. The important part is that it stays upright. So I'm going to place mine in my vessels, and then I have my 3 solvents. So you can just use water at home, or you can use other solvents. I have water, acetone, and isopropanol. So acetone is commonly found in nail polish remover, and isopropanol you might find in your medicine cabinet, depending on what you have in your medicine. You can try all 3 of these. You can try one of them. It is entirely up to you and what you want your experimental design to be. So we're going to take our liquids and we're going to pour them in just so that they cover the bottom of the container and that they actually make contact with the paper. That was the water. I'm going to take the acetone. So we'll check back with these in a couple minutes. Okay, so they've been going for somewhere between 15 and 30 minutes. And as we can see, the colors are starting to mobilize in all of them, but in different ways. So in the water one, you'll notice that the Sharpie marker hasn't moved at all, whereas it has started to move a little bit in the acetone in the middle of the run, or in the middle of the experiment. It actually bled downwards a little bit, which was interesting. And then the isopropanol is moving upwards with the Sharpie. The acetone is showing the most change with the markers. The other thing we can see is that the acetone is moving very quickly. The water is moving at a moderate pace, and the isopropanol is not moving very much at all.
There are many different types of chromatography. What I have here is a non-exhaustive list, but the type of chromatography that we were just doing in the demo is called paper chromatography. The other types of chromatography I'm going to talk about today off of this list are column chromatography, high performance liquid chromatography, or HPLC, and gas chromatography, or GC. So column chromatography is a technique used to separate and purify different parts of our chemical mixture for further analysis. Its stationary phase is called a column resin or powder, and the mobile phase is a chemical solvent. And here I have a depiction of how column chromatography works. So you load your sample in a very small amount of liquid onto the top of the stationary phase. You then add mobile phase, which then moves through the column and pulls your sample onto the column. And the different interactions between the sample and the stationary phase and the sample and the mobile phase separate out different portions of your sample into different bands. And those bands then will move through the column at different speeds, and we can collect the different bands in different what we call fractions. Typically, this is done using multiple different types of solvents because different portions of the chemical mixture are going to have different interactions with solvents of different polarities. Separation is based on the polarity and affinity for the solvent and the stationary phase. So I'm now going to show some pictures of a column chromatography experiment that I did yesterday. So we started off with this beautiful purple liquid. And from that purple liquid, I actually extracted this green liquid even before the column. And I concentrated that green liquid into a very small amount. And I took that sample and pipetted it onto the top of our column resin. What I'm doing here is adding a solvent called ethyl acetate. And ethyl acetate is going to interact with the pigments on the column and start mobilizing or moving some of the pigments through the column. And now I'm adding methanol, which is another solvent, and this is going to move the rest of the pigments through the column. In the end, we are left with 3 fractions, which are the second, third, and fourth vials from the left. And we can see that different colors came out in different fractions. So we have one color in what's labeled fraction 9, which is the third vial from the left, and a second color that is fraction 10, or the fourth vial from the left. The next type of chromatography I want to talk about is high performance liquid chromatography, or HPLC. It uses a stationary phase, which is a column packed with little beads called silica beads, and those beads are all coated in a chemical coating. The mobile phase can be different combinations of solvents. Often it's 2 solvents that are used in a gradient. So you introduce at the beginning mostly the first solvent or entirely the first solvent, and over the course of the analytical run, you decrease the amount of the first solvent and increase the amount of the second solvent until you're left with basically only the second solvent. And it uses high pressure to force the solution or the solvents through the column quickly and onto the detector. HPLC separates compounds based on the solubility in the different solvents and the affinity for the stationary phase. And it's a big, expensive, automated piece of equipment, and this is what one looks like. The last type of chromatography I want to talk about is gas chromatography, or GC. Like HPLC, it's a big piece of equipment. What's different about GC is that instead of using solvents and a solvent gradient, so the changing proportions of the different solvents, we are actually using gas instead of liquid, and we're using changes in temperature. So gas chromatography, its stationary phase is instead of a small column, it's a very long, very thin tube. And the tube has an inside coating of silica with often some sort of chemical coating, not unlike what's in an HPLC column. The mobile phase, like I mentioned, is instead of a liquid, it's a gas. So it's either purified helium, nitrogen, or hydrogen gas. And it separates based on the volatility of compounds and the affinity for the stationary phase. Volatility is how readily a compound is going to move into the gas phase. So we utilize temperature increases to sequentially vaporize molecules. So we are basically picking off one molecule and getting it moving, and then raise the temperature a little bit more, and it picks off another molecule and gets it moving, and that continues and continues and continues over the course of the analytical run. So really briefly, I've mentioned this, but most chromatography systems require detectors so that we can see what is actually in our mixture that we've been separating. Again, this is a non-exhaustive list. In my research, the type of detector that we use the most is called a mass spectrometer, and mass spectrometers are really good at being able to identify what compounds are in your mixture. So all of this has been great, but you're probably also wondering, why do we use chromatography? So like I mentioned, we use chromatography to separate mixtures. For marine science, we're specifically talking about separating mixtures in seawater. And why do we care about that? Well, we want to isolate specific molecules. And we care about specific molecules because those specific molecules are chemical signatures, which can be indicative of different processes. So if we see a specific molecule show up, we know that, for example, zooplankton have been eating phytoplankton and they've been pooping out the remnants of those phytoplankton. Or we know microbes have been munching away at organic matter that's in the ocean. Or we know that a specific type of human pollutant is being emitted into the environment. So we can look at all sorts of different types of processes and all sorts of different types of organic matter to get a sense of what's going on in the environment. Any questions?
Thank you, Paul. You actually answered my question right there at the end with why do you use chromatography and what are the applications can you have? But I do actually have a question about what brought you to this specific field of research and why did you want to study chromatography?
So I actually, I sort of more discovered this as a tool for answering the kinds of questions that I'm interested in. So I'm really interested in how we affect our environment as humans and looking at marine pollution. And this is just, it's a really cool and unique tool that we can use to study our environment and to look at, the ocean is a complex soup of different chemicals and trying to isolate different things to sort of answer the different questions that we have. It's a really cool tool.
Yeah, this might be a silly question, but can you isolate specific molecules using chromatography? Is that what you're looking for?
So with GC and HPLC, the last two techniques that I talked about, they do get with some multiple stages of separation, you can get fine enough separation that you're looking at individual molecules that are eluting at a specific time point. And so you can look at, we actually can get fine enough resolution that the movement of different atoms, so you have the same molecule, but maybe one atom is in a different place. We can resolve those, and by resolve them I mean we can separate them and see the two different versions of this molecule.
That is super cool. Thank you so much for giving your talk. So now
Absolutely.
So now is the time to transition over to Manesh Devana, and he is going to be presenting on seagoing oceanography.
Awesome, thank you. Here we go. There we go. I just want to say thanks to everyone who's helped put this on and all the speakers who've gone before me. Everyone's been super awesome to hear. So thank you. So my name is Manish. I'm going to be talking about what I do as a seagoing marine scientist. So yeah, this is me in a sea survival suit at sea, testing it out, making sure it works. So a little bit about me. I'm a PhD student at Erasmus. I study ocean physics. And so part of our work is we monitor ocean currents and how they respond to climate change. So part of this work involves spending 2 to 3 months a year living and working at sea. And so that's kind of what I'm going to talk about today, is like why we go to sea, what it's like to do work at sea, and then I'm going to get into a little bit about how you actually get into becoming like a field work or a seagoing oceanographer, because that was something I never really, no one told me about. And so I'm just hoping I can pass along what I've learned getting into this field. So why do we go to sea? So this picture here shows kind of a very simple diagram of the circulation across the entire planet. And you can see these arrows go all over the world. And so as part of these currents, they distribute heat and nutrients and biology all over the planet. And about almost half of all of this network is deep beneath the surface of the ocean. And as we kind of saw in one of the talks before, observing beneath the surface of the ocean is really, really difficult for several reasons. The pressure down at the bottom of the ocean, down deep, can reach 2,000 to 3,000 times what we feel at the surface. So that's enough to crush most of our instruments, definitely us if we tried to go down there, so we can't send people down there. And light can't get very deep, more than like 100 meters, a couple hundred meters at most below the surface of the ocean. But we're really interested in what's happening 2,000 or 3,000 meters below the ocean. And then the ocean also has a really nasty habit of ruining basically any kind of instrument we stick in the water. So we constantly have to keep going and putting new instruments and recycling the instruments, because mostly because of the salt just likes to corrode away anything we stick in the ocean. So it keeps us on our toes. And then the last thing, nowadays we have kind of really fancy computers that can model all sorts of cool stuff, but there's still a lot of big gaps in what these computer models can actually do and how they represent the ocean. So it's still really important that we go out and take observations to check what the models are doing and observe what the models can't recreate. So what this kind of all leads to is there's a lot of exploration in the ocean left to be done. And this is one of the reasons I was kind of really drawn and have been more and more drawn to this field as I get further into it, is you really are kind of exploring with every kind of new instrument you stick in the ocean and every new cruise you go. You're observing parts of the ocean that no one has gotten to observe before, and you're mapping parts of the ocean that we've never seen before. So it really is like being an explorer, which for me, that was something that I always loved, like that notion of exploring and kind of being on the frontier. Just like the kind of books I used to read and about like ocean explorers and pioneers, like this, it very much kind of drew me in that way. And so to go to sea, we tend to go on these ships. This is the University of Miami ship, the Walton Smith. There's a whole fleet of US research vessels of all different sizes and shapes that kind of do different things, from going along the coast to deep out into the middle of the ocean. And so we will go on one of these ships and we'll usually spend about a month or so at a time deploying instruments and doing various kinds of work. So one kind of thing we do is we deploy this instrument that's called a CTD. That's this big cluster of stuff going on. And so we use this really cool kind of robotic arm that lowers this instrument into the ocean. So on this big mess of stuff, we've got these bottles that can take water samples, and then we've got a bunch of different kind of sensors that record all sorts of things that are important for monitoring the state of the ocean. And this was like one of the clearest days I've ever seen when we were doing work. So I thought it was a cool video to show. And then we also do work where we leave instruments in the ocean for like a year or 2 years, because some of the stuff we want to measure, we need to kind of monitor on like a long-term basis. So this figure here is showing all these kind of networks of currents that are near the surface and deep in the ocean. And all these little lines are showing where we leave instruments. And this is the work that I do, is really focused on deploying these instruments and maintaining them and using the data from them. So if we like zoomed in on one of these, this is what one of those individual sets of instruments, we call it a mooring, looks like. So you'll have various instruments kind of stationed along it, and then various like anchors and floats. And so every year or 2 years, we have to go out to sea, release these instruments, collect them, get the data, and then put them back in the water. And this ends up being a pretty kind of involved process, unlike the thing I showed before, which kind of is a robotic arm that lowers and lifts it for you. So you basically just have to press some buttons. This mooring work is a lot more involved. So this is a video from my last research cruise out near Iceland. And so as you can see, the boat's moving up and down a whole lot. We were in pretty bouncy waves and kind of 12 to 15 foot seas. And there's a whole team of us kind of moving around, collecting instruments, putting them on this crane here. And then the crane lifts the instruments up, we attach more, and then feed out this big cable. And eventually it forms this long chain of instruments that stays in the ocean for a couple years at a time. So yeah, this is what I spend most of my time doing at sea, and I really enjoy this kind of work because it's pretty fast paced, and you have to kind of think on your feet a lot and work with all sorts of people. So every time you go on a cruise, you might be with local crew from whatever country that ship is leaving out of. You might be with scientists from different parts of the world, and you kind of have to all learn to live and work together because you're all stuck on the same boat for a month plus. And sometimes things go very wrong. So in that last video, we had the crane, but when we were pulling up one set of instruments last time, everything was so tangled, we couldn't put it in the crane. So we had to actually take teams of 5 and pull in 2,000 meters of cable by hand. So this is one shift pulling in all this cable, and these are the instruments and the flotation devices that we all had to pull up by hand. So yeah, this was a kind of a new experience for me. I've done a few of these trips and I've never actually had to pull up anything by hand like that. So that was kind of fun. As you can see, it was kind of cold and chilly. It's a bit of an adventure every time we go out there. But there's also like a lot of downtime. So when you're living at sea, you're literally living there. So you kind of have your morning routine making coffee. That's me making my coffee in one of the computer labs and having coffee out on one of the sunny days. And then we also you find time to have fun. So we set up a dartboard on the last ship. So this is me and Eduardo, one of our coworkers. He's a research tech with us. He's playing darts, and then he's taking a nice nap on one of our midnight research shifts. So I want to talk a little bit about how you get into becoming a seagoing scientist, because this was something that I had to figure out a little bit for myself, because my parents were kind of first generation immigrants from India. They had that kind of this idea of me either being a doctor or engineer, and this idea of being an oceanographer and going to sea was totally foreign to them. And from what my parents understood, it was like a field where people who know how to sail or people who are around boats are the only ones that get to do this kind of stuff. And I like want to start by saying like, that's very much not true. And actually, if you're someone who is not like a sailor and been around this stuff your whole life, you probably have your own set of skills and background that will be an asset at sea and during the travels that are involved to going to sea, because it's not just being on the boat, you're kind of traveling all over the world. You have to deal with people in foreign ports, foreign scientists, foreign crew members. So it's a whole set of situations that you can't predict. So kind of not letting yourself fall into this idea that I'm not someone who like fits into this boating or seagoing crowd is really kind of key. Like you have to kind of realize for yourself, like if that's something you want to do, if you have this desire to go to sea, kind of know that whatever background you're bringing to it will be an asset. But there are a few things that like you should do. So the first thing is study science. It really doesn't matter what kind of science, because it's all involved in the ocean, whether you're doing physics or biology or chemistry or engineering, anything you can think of, you can apply that going to sea. So I do physics. I'm really interested in kind of the physics of the fluid in the like the fluid dynamics of the ocean, but there's all sorts of cool questions about how the ocean cycles different chemicals and how that affects the biology. And there's really cool questions about the genetics of what lives in different layers of the ocean have very kind of different genetic patterns. So kind of any sort of science you think of can be applicable to being a seagoing scientist. And then you just kind of have to ask a lot of questions, not just about like science, but when you see someone going to sea, like a professor who's doing interesting work, like don't be afraid to go and ask them, like, how did you get into this? Or do you have space on your boat on your next cruise? Like you kind of have to be your own advocate and your own, like no one's going to come and find you to do these opportunities. You have to go out there and ask those questions. Can I do this? Will you take me on a cruise? Is there a place for me in this lab? And then just finally, you kind of have to be really curious about everything you're doing and not just whatever science you're studying. When you're out at sea, you're going to have to solve problems that you never would have thought. Like, for an example, we had an issue with some sensors not working, and it turned out there's this like random chemical that they put on the sensor to let it not corrode that melted. So I spent 3 days like Googling what gets rid of that particular chemical so that we could fix it and actually do the science that we wanted to do. And then you have to be persistent. Like going to sea is frustrating. There are days when nothing works and you just kind of have to figure out how to get through it. And there are going to be days when you feel, especially if you are kind of a minority scientist like me, I've definitely had days when I've been out at sea and been the only person who wasn't white on a boat for 30 days. And that can be kind of an odd experience, but I think you'll find that if you kind of are really passionate about the work that you're doing, it'll shine through and you'll kind of make the most of those days at sea, and it'll lead to even better days, and you'll kind of, it makes it worth it. And then lastly, you just want to be ready for an adventure, because at the end of the day, you're going out on a ship out into the ocean, and it is an adventure. And which is why I got into it in the first place, this was this kind of feeling of adventure that I was really drawn to, and it has not disappointed. It has been a whole bunch of things I never expected, but it has not disappointed. So yeah, that was my little talk about going to sea. Thanks for listening. If you guys want to email me any questions, my email's here. I can put it in the chat too. Looks like there's some questions.
Yes, I was just about to ask that. We'd like to know, how long does it take to extract one of these instruments on average with the crane?
Yes, so it kind of depends. So when that long chain of instruments that I was showing, depending on how long of a chain, it can take anywhere from like an hour to like the really long chain. So some of the ones we work with are 4,000 meters long worth of instrument. So it'll be like 4,000 meters of cable with instruments on various parts of it. That'll take almost all day because that crane can only wind in that cable so long. So it's this kind of really long process of like taking shifts of pulling the crane in, taking instruments off, running on. You got to scrub the instruments when they come up, especially in certain parts of the ocean. Stuff will grow on them. So you'll spend a couple hours like scrubbing all this nasty stuff that is growing on them. The biologists who are listening might cringe because they like to save all the samples, but all the physicists just scrub everything off. Sorry. Yeah, but it can take all day to do one instrument, which is why we're out there for so long, because we'll have 15 or 20 instruments to do sometime, or 20 chains of instruments to do.
That is so crazy. I've always wanted to live on a boat and do something like that. So that is really cool. Do you have a favorite memory from one of these excursions?
So on our last cruise, so we were going to Iceland, and because of COVID, normally we would meet the ship in Iceland, and because of COVID, we didn't get to do that. We took the ship from the US and had to go all the way there. And one of the things I was really disappointed was I was missing a chance to see the auroras in Iceland. But one day when I was working a kind of night shift taking measurements, and I went outside to check on the sensors, and the clouds like cleared up, and we saw like the most amazing like Northern Lights aurora out in the middle of the ocean, like just by total surprise. Like we got on the radio, told the captain to turn off all the lights, and everyone ran out. And that was like one of the coolest things. Just being out in the middle of the ocean, seeing the aurora like totally unexpectedly was really cool.
I also hear that it gets really dark out there, so that might have been a very stark contrast, I'm sure.
Yeah, like I couldn't, like I thought my eyes were playing tricks on me at first. And then like sure enough, like it got bigger and brighter and was stuck around for like half an hour. It was very cool.
That is awesome. So I'm looking in the Q&A and you have inspired Kathy. So hopefully she reaches out. Also, Kundan asked, what got you thinking of seagoing scientist?
So actually like I started school as like my college career as a business student. And I quickly realized like I really wanted to do science. And I've always kind of had this affinity for the ocean. And I didn't really know what being an ocean scientist meant, but I knew that I liked science. Like I had like a curious research oriented way of thinking. And so one day, like I kind of, this is where I was talking about being persistent and asking questions. Like I was just kind of emailing ocean scientists from other universities kind of out of the blue. And one was kind enough to talk to me and told me a little bit more about like what it was like to go to sea and what kind of work they did and what kind of questions they were looking into. So this combination of like exploring parts of the ocean and like looking at stuff in the ocean that no one has ever looked at before, combined with this like adventure, like this idea. I mean, I was like the kid who like loved the pirate stories and all the pirate characters. Like that was always something that drew me in. So it was this kind of combination of doing this cool science plus this like kind of inner child in me getting to be this ocean explorer.
That is awesome. Thank you for that presentation, Manish. So we are going to transition to our last speaker of the day, Anna Zongronis, and she's going to be presenting about UF IFAS Extension and Florida Sea Grant.
All right, thanks, Aaliyah. Thanks everybody who's still hanging in there. This has been a long but really fascinating and rewarding day, and it's been humbling to listen and learn about each and every one of the previous presenters, as well as what their field of study is. So I hope to close it out with a bang, if you will, and looking forward to engaging with some of you afterwards. So I am the Florida Sea Grant Extension Agent here in Miami-Dade County. And some of you might be wondering, what is that? I will say that I am a Sea Grant agent, not a secret agent, although that is fun to say as well. And I'm going to explain a little bit about what my job is shortly. But first, I wanted to go into a little bit of my path and how I got here. For those of you who were on earlier this morning, we heard from Stephanie Wright, who talked about her background in the arts and that she had been a theater major and a drama student. And where I am now is actually a career change for me. I used to be a working news photographer. I have 2 undergraduate degrees in photography, and it was my #1 dream in life to work for a major metropolitan newspaper, which I was very fortunate to do in central New York for a number of years. And unfortunately, that fizzled out at the end because of the internet and a lot of media going to online formats that weren't paying. And so, that industry changed quite a bit. And so I retired and then started looking for my next step. And at the time, I had moved back in with my parents. I was almost 30 years old, boomerang child, and was working a number of part-time jobs to support myself and my pit bull. And one of the things that I was doing that brought me a great amount of joy was that I was serving as a volunteer scuba diver at the National Aquarium in Baltimore, Maryland. And this brought me back to my roots from when I was a kid, when I wanted to grow up and be a marine biologist and train killer whales and dolphins. And so this brought me closer to that in a different way and really had me looking at the underwater world through a different lens. I had been a diver at that point for 9 years, and actually this month or March will celebrate my 21-year diving anniversary. So that has always been a major passion and led me to pursue going back to school at Rasmus for the Master of Professional Science degree. And for those of you who might not be familiar, this is a slightly newer master's degree track that varies a little bit from a traditional Master of Science or Master of Arts in that it's an accelerated program. And at the end, rather than doing research, the student participates in an internship, which is serving either a larger agency or organization. And this appealed to me a lot because, as I said, I was already at this point, I was in my early 30s and switching fields. And so the idea of practical work experience was something that was very appealing and I thought pretty relevant to me because I wanted that on the ground experience. I was really lucky to land a longer internship following my master's studies. I had a 10-month internship at Biscayne National Park, which is based out of Homestead, south of Miami. And once I finished that internship, I secured another and then got to work there as a National Park Service employee and wear the flat hat. And I was an interpretive ranger for a few months. After that finished, I somehow, you know, they say that life happens while you're busy making plans, but in my case, it lucked out that I was able to apply for a position at the Florida Department of Environmental Protection's Coral Reef Conservation Program, which is based here in Miami. So that was my first full-time job after grad school. And similarly to what Stephanie mentioned earlier, that all of her experience and in her background really prepared her for coming to her new field of study, I can definitely attest to that. My experiences growing up as a photographer and then working at a newspaper and photographing weddings really prepared me for being out in the public and working with the public at the Park Service, even though I was working as a biotech in my internship. And then when I was working at FDEP, that was another position that really required a lot of public engagement. So being able to communicate science effectively and efficiently was an incredibly important skill to have in that job. After a little over 2 years at FDEP's Coral program, it was time for me to move on. And that's where I landed where I am now. And that's an employee. I'm duly employed by the University of Florida IFAS Extension Service and Miami-Dade County. It's a state-county partnership in which I have one job, but two bosses, if you will, and I'm paid by both. And it's not that I get paid more, I get paid my salary, but I get two paychecks that total up what I earn. And so, you know, if that didn't confuse you, hang on, I'm going to throw more at you. In my role as a Florida Sea Grant extension agent, I am representing the Florida Sea Grant College program, which is part of a national program of 34 academically based programs that communicate science to marine and coastal stakeholders specifically. So stand by, hopefully I'll help clear it up, you know, coming up. So two bosses, but I report to three entities, and I will explain what I do coming up. University of Florida IFAS Extension is where the state of Florida's Cooperative Extension Service is housed. And University of Florida, I'm sure some of you are familiar, the main campus is based in Gainesville, and I am extension faculty of the university. An extension means that I am a branch of the university, but I am a local branch. In other words, I am a Miami-Dade County-based faculty member. And the mission of UF IFAS Extension is to partner with communities to provide quality, relevant education and research-based information, so scientifically based information, to essentially bring a balance between healthy people, healthy population, healthy economy, and healthy environment. And this happens a number of three ways, or I shouldn't say a number of three ways, a number of ways. So in essence, as an extension educator, it is my job to teach. And I do this through workshops and classes and trainings, webinars, but also things like publications, videos, blogs, and social media. And our three guiding principles, if you will, are: the first is that it's our job to teach people stuff. So our first or short-term outcome of our work is knowledge gain. We need to be able to show that people have learned something. Taking it a step further, a more midterm outcome is behavior change. So people learn stuff, and then after the fact, what have they done as a result? How have they created a new behavior or changed a habit that will result in one of those outcomes of healthy population, healthy economy, healthy environment? And then longer term outcomes that we're aiming to capture include the acronym C, or social, economic, and environmental impacts. So everything that we do we start at the end and work backwards. So in other words, when we're determining what it is we're trying to achieve, we look at that long-term goal. What is it that we need to do? And then start backwards, building forward from there. And part of this includes building in a way to evaluate what we've done, how effective it was, who we've reached, etc. So it's not just a matter of teaching people stuff and then sending them on their way. We have to continue to maintain contact and reach back out to them to fill out a survey and see what they've done 3 months after they took our class, 6 months, a year after, etc. So kind of like those surveys you get when you fly JetBlue or American Airlines, how was your experience? It's like that, but it actually is really important to us. And as I mentioned, Florida Sea Grant is the marine science component housed within UF IFAS Extension. And our motto is science serving Florida's coasts. And it's our role to deliver this best available science to our stakeholder groups in ways that people can understand and apply into their recreational activities or their work activities, or both. And here in this map, you should be able to see roughly all of the green areas reflect a Florida coastal county that has a Sea Grant faculty member. So here I am down in Miami-Dade County. We also have an agent in Monroe County, and pretty substantial coverage throughout the rest of the state. There are cooperative extension offices in each of the 67 Florida counties. So if you're interested in cooperative extension or are curious about what other programs exist, I will be putting my email address into the chat. So my focus are the marine and coastal resources of Miami-Dade County, and regionally a little bit larger besides Miami-Dade County and our state. And as such, all extension agents are responsible for deciding how our work will be conducted. And so we have to create or establish what are known as our capital P program areas, or in other words, the themes or broad general topics of what we do will fall under. And in my case, my two programs are promoting healthy coastal ecosystems through restoration and citizen science initiatives, and enhancing marine and environmental literacy and stewardship. All of our work as extension agents is extremely client focused. As a Florida Sea Grant agent, my audiences or key stakeholders that I'm looking for, that I look to work with, are those that are very dependent upon the coast or the marine resources. either for their work or recreation or both, as I mentioned. Here on the screen, you can see this is a pretty good representation of whom I work with. And there are times where there'll be this silent category, if you will, of like the community at large or the general public. But for the most part, all of the work that we do, all of the extension education that we conduct is geared toward a very specific group. So it's pretty clear to see that these are all folks who are very reliant upon the water. What does a Sea Grant agent do? I thought it would be fun to include some photos. There were memes that circled a few years ago, like what my family thinks a marine biologist is, and it's a picture of someone kissing a mammal, and then what I actually do. So there's always the myth, the man, the myth, and the legend associated with our jobs. So I do have the opportunity to get some glamour shots, like you see here, when I get the chance to do some field work. And in these cases, I will do field work either for my own projects, or I'm assisting partners or other organizations to further science in Miami-Dade County. By the way, in the photo in the center, I am not throwing up, although if I were on that giant ship with Manesh from a few minutes ago, this is what I would be looking like if we were in 12-foot seas. I'm actually bending over the side of the boat to collect a water sample. So as extension agents, the majority of what we do is computer-based or administrative, and that we are planning and executing and delivering our programs. And this involves designing those end objectives, designing the educational materials, advertising to the specific groups who are looking to engage, and of course, designing that evaluation metric. So 75% to 80% of what we do is planning. And I would say 20 to 25% is the actual execution or delivery of our extension programs. So this is more realistic, or I should say, traditionally what my job looked like. Now it's very similar, but it's a little more Zoom based, but it's starting to get a little bit back to in-person activities as the COVID pandemic continues to evolve. But I work with people, and that's something I also wanted to highlight that I really loved about all of our presenters, is that they really emphasize their particular connection to their career path and how they grew up and what influenced their paths. Because at the heart of everything, we are people, and we have to connect with other people and try to be respectful of other people despite shared positions or interests or viewpoints. And I'm really fortunate in this job that that's part of what I get to do, is connect with stakeholder groups consistently. I wanted to give a brief highlight of some examples of the different types of my projects and how they connect to that extension model of knowledge gain, behavior change, and the C. So the first is that knowledge gain, which at the barest level, we hope that no matter what people do, whether they're reading a publication or watching a video or reading a blog or taking a class, that they are learning something, they're learning stuff. And the majority of the people I work with are adults. There are things that I do with youth, but my target audience are adult marine resource users. And that's largely because here in Miami-Dade County, there are a lot of nonprofits and other local organizations that have a ton of youth programming. So part of what I need to make sure I do is try not to offer something that's duplicative. I don't want to create competition when there isn't really a need to do so. I want to highlight what my partners are able to do and highlight what I bring to the table as well. So one example of knowledge gain is my teaching of the Florida Master Naturalist Program courses. And the photo of the left is a screen grab. I'm sure we all have pictures like this, either from online classes or family meetings that we've held through online platforms. This was our first virtual Florida Master Naturalist Program course last year. And you might be able to see in this screenshot, some of the students got really into it, and they were able to put little graduation caps on their heads for the class picture, which was really fun. And on the right This is a photo of one of the classes on one of their field trips in which we were having them bag up oyster shell that we've been recycling in Miami-Dade County, which I'll touch on shortly. Also following in line with the idea of knowledge gain are citizen science projects that I've had the pleasure of working with here in Miami. The first was an observer training that my colleague and I created to engage recreational divers in stony coral tissue loss disease response efforts. And so this program had two solid objectives, and that was they were to increase divers' understanding and awareness of the coral disease outbreak that has been happening ongoing in Florida since 2014, and also to expand the underwater surveillance network that we have here in Florida, because as you can imagine, we have a lot of water and a lot of reef, but it's particularly challenging to be able to monitor all of that area because it relies upon a very particular group of individuals who have that capability, and those are divers. Additionally, I took over and ran a citizen science boat-based water quality monitoring program in Biscayne Bay called Biscayne Bay Water Watch, in which participants went and monitored a particular site for the same parameters that Miami-Dade County has been collecting for 30 years. So this is one of our longest and most intrepid volunteers, a fisherman, gentleman in his 70s, who would go out on days that were not like this one, not calm and flat, but have 2-foot seas in the bay, which is a lot, on his little flats boat, and then come back and tell me about it. And I would always say, Herman, if the day isn't safe, you don't need to go out. And he always just had to go out, it was pretty fun to have those interactions with him. The current and newest citizen science project that I am taking on is the expansion of Florida Horseshoe Crab Watch here in Miami-Dade County. We had our first training event a week ago today at Crandon Park, and we had a wonderful turnout within the UM Rasmus community. This is the photo of all the UM students that came and participated. And this program is looking to survey and help establish population targets for horseshoe crabs here in Miami-Dade County. This is a statewide program, but this is the first time that we're going to have active surveillance for horseshoe crabs at particular periods in the county. So again, if this sounds like something of interest to you, please reach out. I'll be throwing my email in the chat. Now, the next tenet of the extension model is behavior change. So what did people do with all of this great stuff that they learned? A couple of examples that are pretty easy to remember are, one is that I do a mangrove trimming workshop for landscaping and arborists, the professional landscaping and arborist community. If you're not familiar, mangroves are incredibly valuable trees, both ecologically and economically in our state, but they're also very highly regulated as a result. So individuals who can trim these trees have to maintain certain licenses and comply with all sorts of regulatory rules. And so part of our workshop is teaching the proper trimming techniques and also bringing in representation who can help answer a lot of those regulatory questions, because no one can remember all the rules. And sometimes people, even when they mean to do the right thing, sometimes by accident, they might violate something. So we're trying to minimize that and get this type of professional development for this particular community. Additionally, our work with stony coral tissue loss disease has expanded outside of Florida. And one example in particular is our work in Puerto Rico. We traveled to Puerto Rico a little over 2 years ago to teach a workshop before this coral disease was detected there. And since then, the disease has established around Puerto Rico, and one of the stakeholders who participated in the workshop has continued communication with me, and now she holds the permit to perform coral disease treatments in Vieques, Puerto Rico, which is just off the main island. So that's a pretty big win for us. And so now she is the one treating all the corals in her particular part of Puerto Rico, which is really exciting. And lastly, if I haven't lost too many of you, I'm going to give a brief example of those social, economic, and environmental impacts. And the example that I'd like to talk about is the oyster shell recycling program that I started with another colleague here in the county, in which we contacted local businesses to collect their discarded oyster shell. Now, the eastern oyster, that is the kind that is edible, it's no longer naturally occurring in large populations here in Miami-Dade County, but restaurants bring it in, they purchase it, and the shell that is discarded can actually be used by other counties or other regions of Florida in restoration projects. So after we collect the shell, we work with 2 or 3 local restaurants to collect their shell. We cured it outside of our research facility down in Homestead. And we had an activity with our Florida Master Naturalist program to bag up the shell. And after that, it was sent up to Cedar Key for restoration. And Cedar Key, if you're not familiar, is up in the Big Bend area on the Gulf Coast of Florida. And they took our shell and placed it into these reef prisms, which is a newer technology. It is a plastic-free technology for oyster shell restoration. And our shell was enough to create 66 reef prisms, and it was used to restore approximately 500 linear feet of shoreline in a Living Shorelines project in Cedar Key. And that oyster shell restoration is expected to deliver about $9,000 of ecosystem services to the area. And I'm starting to wrap up. I'm getting in the home stretch, but I wanted to give you guys an example of some of the figures within the citizen science and volunteer work that I have collected over the years. And if you don't have to worry necessarily about everything, but just look at the very bottom to the highlighted figures. And you can see that we've worked with over 200 volunteers. And this is just within my program in Miami-Dade County. And all of that oyster shell that we recycled was almost 5,000 pounds and almost 6,000 pounds of marine debris removed. And the University of Florida associates a dollar amount with every volunteer hour that is contributed. So the cumulative amount of volunteer time associated with what you see on the screen is $22,293, which is huge considering that a lot of local agencies and organizations are very limited in manpower and funding. So that makes these volunteer contributions that much more impactful. I also would like to mention that none of these activities happen in a vacuum. You know, there are people who have the great ideas, and then there are people who help execute them. And so that's what partnerships are all about. And that's where relationships are really everything in the marine science field. It's a very small field. So often as you go out and you do things, you'll meet someone who you were on a call with or a project with a few years ago. It's a very tightly knit world. And leveraging those relationships, not just for your own benefit, but also for the benefit of your partners, is really critical. And none of the work that I do would be possible without partnerships. And these are just a sprinkling of these logos that you see on the screen are just a sprinkling of the partners that I work with. So that brings me to the very end. And I want to thank you all for your attention and hanging in there. And I invite you to keep in touch with me and ask questions.
Thank you so much, Anna. So it looks like you just answered the question in the Q&A, but do you have any recommendations for volunteer opportunities that anybody can get involved in?
Definitely, that's a perfect question. So again, between myself and other partners, I'll try to throw those in the chat if you all don't mind staying on for a minute. But Frost Science Museum, they host restoration opportunities at Virginia Key North Point Park, which involve removal of invasive plants, as well as planting of native vegetation on the dune system there. That's a great place to go. And it's really rewarding because you can see immediately what you've done and how it's changed the landscape. Additionally, volunteercleanup.org is a fantastic website. The organizers are based in Miami Beach, and it's a platform that lets you look for and participate in marine debris cleanups around the county, or host your own and use their platform to recruit participants. So again, that's volunteercleanup.org. And they are also the Miami-Dade County coordinator for the International Coastal Cleanup, which happens in September. I would be remiss if I did not mention the Baynanza event, which is coming up in April. Baynanza is a celebration of Biscayne Bay, and this year is its 40-year anniversary. Don't quote me on that, I could be wrong, but it's a big one. It's an anniversary, and that is hosted by Miami-Dade County and a number of partners. So there will be educational events as well as bay cleanup events throughout the county on that day. And I would also mention Debris Free Oceans does a lot of wonderful events. They are largely work out of Miami Beach, but they do other things throughout the county as well. So there's a lot. I'll put them in the chat.
Do you have anything in the Orlando area? I know that's kind of a landlocked area.
I don't specifically, but I can put you in touch if it's either Orange County or the other O County up there. I can, if this person will shoot me a note, I can connect them with their local UF-IFAS extension office, because that office would be able to better guide them in terms of what opportunities are happening there and what's available locally. I'm happy to make that introduction. Osceola, Orange or Osceola County.
Yes. So just so anybody doesn't have a question right now, you can always send Anna an email later on if you come up with a question or anything specific that you would like to ask for help with. She is also one of the hosts for the event and will be able to answer any questions afterwards. So at this time, I would just like to give a word of advice to those looking to pursue a career in science. We had so many great speakers today who spoke about their backgrounds and how they came to be where they are today. But I guarantee you that each and every one of these people have experienced hardships and obstacles that they had to overcome. Don't let those obstacles that you are guaranteed to encounter in this field discourage you. Okay, science is hard, but it is super rewarding. I know for me that it gave me a sense of purpose in life, and no doesn't have to be the end. Okay, keep fighting for yourself, keep advocating. It's never too late to start, and you do not have to have all the money in the world to do this. You do not have to have experience. You do not have to have the perfect GPA. I know that's rich coming from me, someone at Rasmus, pun intended, but I am here based off of scholarship and really, really hard work. And I'm going to share with you a short story about my background where I was previously ashamed for a long time. I remember walking into my advisor's office in undergrad, wanting to get into the fisheries management field. And I had to tell him, I have a 0.5 GPA. What do I do? And yes, you heard that right. I said 0.5 GPA. That means I had one A and all the rest were D's and F's. It was a lot of work to get where I am today, but I had to fight. I had to pull up those grades. I had to find something I was passionate about. I started in a field I was not passionate about, and I had health issues that put me out of school for quite a while, to be honest. But don't stop advocating for yourself. Don't stop taking up space. And if there's only someone, if there's no one that looks like you, that is okay. That is completely okay. I may have had poor grades. I may not have had the time or money to do the internships that I wanted, but I worked hard. Use what you may consider to be your pitfalls and change those into stories of success. I got here with lower GPA than others. I pulled it up to a 3.0. It was 3.02. But you know, you can do it. Keep fighting. And also find mentors. Science has historically been exclusionary. But at least in this community, I've found that there's a lot of advocacy and work being done behind the scenes to create an inclusive environment from those who have been underrepresented and historically excluded from these spaces. So please take up space. I would like to take this time to thank all of our speakers who came out today, took their time out of their Saturday, and all of our guests here as well. Thank you so much for encouraging these future scientists and sharing your wonderful stories. On the screen, you'll find a QR code for those who are still with us, as well as a link to fill out a survey. And this feedback will really help us in future years to see what you would like to see in the future. So thank you so much.